Right. Well, good morning, everybody. I'd like to thank all of you for taking this next step in your careers to become certified. This has been a great career for me. This will be I guess I'm in my 34th year of doing these sorts of things. My background is I have started at the very bottom, dragging brush. I've done everything from that to crane removals, to plant health care, to consulting, to construction mitigation work, to education. And I still am the owner of a plant healthcare company and a consulting company, business to business type. So I want to take the opportunity now to begin the tree biology section. This is the foundation of most of the other chapters within the book. When I first took this exam decades ago, the tree biology section was carried the most weight. Now that's no longer true. However, it is still very foundational to the rest of the chapters and your understanding of everything about pruning and stress response and things like that. So, sure. All right. So if you have any questions or comments, this does go on for 7 weeks. Please feel free to reach out to me if you do have questions or need a little bit of guidance. You can see my email address here. Try to put something in the subject line so that it doesn't go to my junk email folder. The certification guide that you should be using should be one of the newer guides. And I also highly recommend that people use additional, let me see if I can get my laser pointer going, some additional resources. The exam itself not only comes from this study guide, but it also comes from experience, and it comes from our safety standards and operational standards. We operate under the ANSI Z133.1 safety regulations. These have changed a little bit, but we'll go over those when we get to them in the safety chapter. We also are covered under OSHA, so OSHA and ANSI, as well as the Arborist certification guide. If you follow all of these, you should have an easy time of the exam. The Arborist, so this is the newest certification guide, the 4th edition. This is the one we are currently using. There's not a whole heck of a lot of difference between this one and the previous edition, but there are test questions that will only be answered from the 4th edition. Here's that extra set of standards that I spoke about, the ANSI A300s. They are in process, if they have not already done so, of combining all these standards into a single standard, the A300. No longer will you see individual parts 1 through 10. It'll all be a single encompassing standard. But these may look familiar to you. These are all fairly similar to what you see in your study guide as well. However, these are the primary source. I always like to go to the primary source to get another perspective on the same information, much like why you would come to one of these classes, so that you can get a little personal interaction with it. Okay, so we'll go ahead and jump into the biology section now. We're going to start off, we'll talk about cell structure and a little bit about its composition. We'll talk about meristems. We'll talk about differentiation, which gives rise to things like buds and leaves, wood and roots. The rise of the vascular systems, mycorrhizae and the development of the root system, photosynthesis and respiration, tree growth and development. We'll talk about compartmentalization of decay in trees, or the CODIT model. And then at the end, we'll talk about some palm anatomy and physiology. What actually makes a tree? What's this definition of tree? Trees are large, woody plants with a single or multi-stem trunk. They're long-lived. They're compartmentalizing perennials, meaning they persist from year to year. Why do we need to know how a tree grows? Why do we need to understand tree biology? Well, in order to know if things are broken or not, you have to know how they're supposed to be, how things are supposed to respond. And so with a better understanding of biology, you'll know what's good and what's not so good. Also know that we'll understand how genetics play an influence on how trees appear and how they behave and interact with their environment. So we'll talk about the component parts of the tree, anatomy. So anatomy is the science of the structure of animals and plants. Morphology is the study of the forms those things take. What do they actually look like? Physiology is a function. So things like biological processes, physical and chemical processes. Cells are the basic building blocks of tree structure. New cells arise from the division of existing cells. In trees, division occurs in specialized zones called meristems. So there's several places we'll find meristems within the tree, but that's where the differentiation occurs. That's where new cells are manufactured. And we'll talk about where they stay in a second. But so after cells divide, so you have a parent cell, it divides, and you get a new cell, they can undergo differentiation. So it can go from sort of a generic tissue to something with a very specific function. And usually that has to do with wherever the cell is located at the time that division occurs. If it's occurring in a root, the cell is likely to become a root cell. If it's in a secondary meristem, it's likely to become more vascular tissue. If it's inside of a bud, it's going to become part of a leaf or a flower or some fruit, something like that. So it's wherever that cellular division occurs, very close to that, it will take on the habit of the adjoining cell. But does it have to? No. You can cause these cells to become other things, which, if you go into science and you study this stuff in college, you'll trick these cells into doing just about anything that trees can do. Cells with similar structure and function are arranged into tissues such as bark and wood. So cells give rise to tissues, and tissues give rise to structures. So if I'm a epidermic cell on a leaf and I'm producing more and more of these, I create the covering to the leaf. The adjacent tissues are doing the same thing. So eventually you get a whole leaf, which would be an organ. So here we have the main organs of the tree: leaves, stems, roots, flowers, and fruit. Together, these form a fully functioning tree. You can see our diagram. Throughout arboriculture, you will find out that there are multiple names for each part of the tree. I try to use the terminology that's going to show up on your exam. Another thing that you need to keep in mind is that these exams are international. They are not just the Piedmont region of Georgia. They are meant to cover trees in the temperate forest ranges around the entire world. We will discuss tropical trees and maybe even a desert tree or two throughout this lecture. However, we are primarily focused on the forests, the temperate forest regions of the world. But as you go around, even within the United States, you might hear things like, you see where it says bole here. Some people might refer to that as a spar, or even on here, it'll say trunk. So there's a number of names for each one of these things. Just keep that in mind. So I talked about meristems. Meristems are where that differentiation occurs. That's where that cell is developed. That's where the growth happens. And we divide that into two types. We have primary growth and secondary growth. Primary growth has to do with the tree getting longer, or the branches getting longer, or the tree getting taller, or the root system getting longer. So that it has to do with elongation. So primary growth has to do with elongation. The growth of these shoots and roots occur at apical meristems. So apical means like the apex, the top, the most dominant. So, and trees are compartmentalized, so a branch is independent of the main trunk. So you have apical meristems up at the very top of the tree. You might have an apical meristem out at the end of a branch. You'll have an apical meristem at the end of the root. So just think of things that are getting longer as the primary source of getting taller or wider or more pervasive when it comes to the root system. Then you have secondary growth. Secondary growth, is the bulk of cellular division within a plant. This is it getting larger in diameter. So it's not length, but diameter. So secondary, this, if you look under the bark, we've all heard about cambium tissue. Hopefully you've read through this material before you've taken the class and you're understanding these terms. But cambium is just under the bark. If you peel the bark off and you feel that little slick layer right under the bark, that's likely to be the cambium. And that's what's going to produce our vascular tissues and our cork. So there's two types of this secondary stuff, and that is vascular cambium and cork cambium. Meristems within the trees, branches, and roots. So no matter where you are on the coating of a tree, you will find cambium tissue. They produce the woody part that remains, all that xylem tissue that builds up year after year. It allows trees to grow quite large. But here's a key factor to keep in mind. Notice it says palms lack secondary growth. And we will put this to practice when we get into the pruning section. We'll also go over it a little more here to understand the biology of palms, but it's very important to keep in mind that palms do not have secondary growth. So let's take a look at these a little bit closer. Here is a root under a microscope. Right in this tiny little center right here, this is the apical or the primary meristem in this root. On the outside of it, this is what we refer to as the root cap. This root cap is a harder material. It also produces an exoate, the protein that is like a lubricant that can help push its way through the soil and exploit the soil. But it's pretty durable so that the apical meristem can continue to do what it does, and that's to produce cells. So you'll notice that these cells are pretty small right around here. They get a little bigger back behind it. And so you're going to have to put your thinking caps on just a little bit, and I'm going to ask you to think over in terms of 3 dimensions plus time. So I guess all 4 dimensions. So this is going to be a fixed point in space. Here's my apical meristem today, right now, at this minute, and I'm going to continue to grow. And as I grow, I'm going to produce cells right behind that meristem. So as I grow along, that cell that's produced doesn't move. The only thing that's moving is that root cap, and it's pushing its way through the soil. And the little meristem, the little apical meristem, is moving along, producing cells back behind it. And those stay wherever they're manufactured. So they're fixed in 3-dimensional space. Hope everybody sort of can see that. That'll also become an important axiom about trees as we get into pruning. So the lateral meristems, again, lateral is this way, so it's getting larger in diameter. And we can see that at the very heart of the tree, if you cut a tree down and you look on top of it, you will see those tree rings. Those consecutive rings are old xylem tissue. So last year's xylem is abandoned as the new year's xylem is being produced. And it doesn't necessarily die. It does over time, but it might take 10, 15, 20 years for that xylem tissue to totally become heartwood tissue. So it can stay alive and useful for quite some time. So to the outside, we're going to have the phloem. So we have this very thin, here's the cambium. The cambium is a very thin, continuous tissue growing around the tree. Again, it's producing xylem tissue to the inside, phloem tissue to the outside, and then on the outside of the phloem, we have bark. So here's sort of an example of the two types of vascular tissues. Here's the cambium tissue for the vascular cambium. Again, this direction is towards the center of the tree. This direction is towards the bark. This thin layer of the cambium producing phloem tissue here and xylem tissue here. Over in the cork cambium, we are producing a phyloderm to one side, and then to the outside, we have the older corky cells, the epidermis and the cuticle. All of this old phloem, because its phloem is not accumulated year after year like the xylem tissue is. The older phloem tissue breaks down and is incorporated into the cortex of the bark. That's kind of recycled. The plant cell walls. This is an important concept that separates plants and animals. Plant cells have hard cell walls. Animal cells do not. If you get some type of injury, you personally, that injury is repaired. If you have a cut, that the wound will close. The cells that were damaged are scavenged and are replaced with living cells over time. You might wind up with a little scar, but it's all new tissue. Plants do not have that ability. Once a plant cell dies, it's dead, it's not replaced. And those cells, again, are fixed in three-dimensional space, so they're going to stay wherever they are. They're not absorbed at all. So that dead cell is going to remain. Cell walls are made up of primarily cellulose and lignin. Cellulose is the structural component. This is what gives it its morphology, its shape, its form, its appearance. It's the most common organic compound on Earth, and it provides the architecture of different types of cells. Lignin adds strength and rigidity. This is what allows it to bend. This is what allows it to absorb compressive forces, and it allows the trees to grow very tall. Let's talk into the vascular tissue now. The primary functions of xylem tissue. Remember, this is that woody tissue that's going to persist year to year. The current year's xylem tissue conducts water and dissolved minerals. It supports the weight of the tree. It stores carbohydrate reserves and defends against the spread of disease and decay. It is a complex of tissues. Whenever you see in arboriculture the word complex being used, it means that it's more than one thing going on. And here we have two types of tissue, even though it's two types of function of the same tissue, the xylem. We have symplasm and apoplasm. Symplasm is the living part. So if you were to cut it down and you look on top, you cut down your tree and you look on top of it, you see that sort of bright white section, and then towards the center of the tree, if it's big enough, you might see some darker wood. That bright white stuff, that's all alive, and that's the living symplasm. The darker part is the apoplasm, and it's non-living tissue. Most of the tree is going to be made up of, at least a mature tree is going to be made up of this type of tissue. It's important to remember that in that live area are stored carbohydrates. So there are a few different formations that happen inside of the xylem. If we were to look at this picture, this is the end of the previous growing season. This would be spring. As you go through the growing season, cambium's constantly producing new vascular tissue. And so these are older cells than these are at this side. So as I go through spring, you see that these tissues are fairly large. And as I get towards the fall, when the sunlight gets less and less, the temperatures continue to drop, the size of the cells gets smaller and smaller. And then we go into winter dormancy right here, and then a new year begins on the other side of that. And then you see a couple of structures like this one that runs all the way across these annual accumulations. And then inside of it, during the annual accumulations, you see these larger things. So these are, in this case, we're talking about a gymnosperm or a conifer. We have tracheids. These conduct water and provide mechanical support. They are elongated dead cells with pointy ends and thickened walls. So they kind of overlap. So one tracheid cell's here, the next one will be here, the next one will be here. So the has to kind of make this little zigzag. Xylem tissue carries things from the root system up into the canopy. So this is where the tree's going to pull its water and all the dissolved minerals and the things that it needs for photosynthesis as well as protection. There's also going to be some fibers. So you'll notice that there's this sort of hole in the middle. So that's the tracheid. And then you see this discolored area around it. These are the fibers and the parenchyma that are going to sort of hang around. You'll also see parenchyma cells along these things called ray cells. So these fibers, again, mechanical strength, the parenchyma, these are living cells interspersed among other xylem cells. They store carbohydrates in the outer layers of the xylem. In other words, the newer part, the live part, the symplastic part. They defend against decay. We'll show how that happens a little later when we get to CODIT. And they have a structural function across the grain. So again, here's that cross section of the pine showing the parts of 3 annual rings, 1, 2, and 3. The one complete growth ring in the center has early wood to the right, late wood to the left. Pine, a gymnosperm, has no vessels. We'll talk about vessels when we look at hardwoods. The wood's rays are 1 cell wide and run horizontally across the image. So these are the ray cells right through here. There's another ray cell running across. Let's take a look at an angiosperm. So these are the hardwoods. And so here we have trachea. We've talked about tracheids, fibers, parenchyma. Parenchyma is going to be a lot more abundant in hardwoods and arranged close to the vessels. Vessels are the primary conducting elements. They are stacks of dead hollow cells that form long tubes. So previously we talked about tracheids. So tracheids are going to be, you should think about those in terms of conifers, and vessels you should think about in terms of hardwoods or angiosperms. Vessels again are the primary conducting elements. So in the tracheids, you had this sort of dead cell and then another dead cell and another dead cell. So they don't stack up end to end. In the hardwoods, the vessels stack end to end like a continuous straw. And again, it's important to know that these are stacks of dead hollow cells. So put your thinking caps on again. What does that mean? That means that water has to move up that tube under some magical force, and we'll talk about this magical force a little later. But because the cell is dead, there's nothing there that can contribute to the movement of water. There's no energy source to move water inside of these tubes, and we'll get to that eventually. These tubes are more efficient than tracheids in conducting water, so they can respond much quicker than a conifer can to rainfall. Now, in these hardwoods, we can divide hardwoods vascular tissue into 2 arrangements. We have ring porous and diffuse porous. So ring porous trees are elms, oak, ash, and you can look these things up. Feel free to go out there and Google whatever tree it is that you have in your area to find out if it's ring porous or diffuse porous. Or you could when you're pruning, you can take a branch, cut it, look at it under a hand lens even, and you'd be able to tell the difference. You do not have to necessarily use a microscope to see the difference between these 2. Ring porous produce wide vessels formed early in the growing season and narrow vessels formed later in the season, much like early wood and late wood or spring wood and fall wood. Diffuse porous vessels are uniform in size throughout the growing season. And if you look at these trees, you can kind of see a trend, and it doesn't always hold true, but it might help you to think about the difference in ring porous and diffuse porous. Ring porous trees tend to be upland trees, trees that grow on slopes, trees that might not have a constant supply of soil moisture. But again, it doesn't always hold true, because here you have an ash tree that might exist in both locations. Diffuse porous wood typically occur in areas with constant soil moisture. So there's no need to have extra large tissue in the spring to take advantage of the rainfall, and smaller tissue later in the year to protect itself from drought. You might not need that extra bit if you're growing in an area with constant soil moisture. So you get this diffuse porousness. So here's an example. Again, we have 3 years represented in this picture. Here's the beginning of the year. Notice the big vessels, and then as you go through towards the end of the year, the vessels get a little smaller and more compact. This would be ring porous. Diffuse porous, the size of these things are relatively the same from beginning of the year to the end of the year. Notice in this one, it's a little harder to tell. You see the line, so we know where the tree stops growing, but we don't necessarily know in which direction these things are going. Also notice that there's these little blue stains scattered throughout. You know, that blue staining is a reaction to being damaged. So when a tree gets pruned, it wants to wall itself off, and we'll talk about that in the CODIT model. But one of the ways that it can inhibit the movement of a pathogen is by plugging the vascular tissue with a substance called tyloses. It's a naturally occurring polymer. So that's what happened. That's why these things are blocked up. So if the tree had not responded to the pruning wound, these vessels would all look much more similar. So they're in size. So again, diffuse porous and ring porous. Here's an example of the heartwood, the apoplasm, and then out here, the symplasm, the sapwood, and the heartwood. Again, if you guys have questions as I'm going through these, feel free to put them in the Q&A, and Brittany will get a hold of me to let me know that you have a question. If you want me to rephrase it or try to add more to it, I'm happy to do that. One of the drawbacks to the online classes where I can't hear the feedback is I can't drill down on what it is that you don't understand or you don't know. So I have to present it in sort of a very generic, all-encompassing format here. But I do want you to ask questions as we go along, because not everybody understands or is taught the same way. So please use that feature. Now, growth rings, we talked about this being an accumulation of aging xylem tissue. So the growth rings are found in the xylem. The rings themselves are formed by the cyclical nature of a year. So we have fall, spring, most of the growing season, another fall, then we have winter at the line, and then it starts over again and goes over and over, forming these sorts of rings. It's only visible in a cross section. It's the result of the seasonal production of xylem by the cambium. Now we talked a little about early wood and late wood. The early wood, these are the cells that are produced early in the season, and they can take advantage of spring rainfall. Late wood, these cells are produced later in the season. They tend to be a little smaller. The early wood is starting to get shut down from moving things up and down the trunk. And so the tree's better able to defend itself against drought by making late wood. Conifers do not have vessels and are therefore non-porous. Growth rings may not be annual for tropical trees. Again, the closer you get to the equator, the less difference there is between summer and fall. And if you don't have a winter, you never stop growing. So you don't have this sort of dormant period in the tropical trees. Therefore, you would not have growth rings. And again, palms do not form rings of xylem tissue at all. We'll Again, palms are their own special breed. My favorite plant. No, just kidding. They're not my favorite plant. But so if we go back to this porous and non-porous thought for just a moment, ask you to think about some hardwoods. Talked about this tyloses that's produced here. Tyloses, some species produce tyloses without injuries, such as the white oak. So as the white oak ages, its older vascular tissue, like the previous year's vascular tissue, is all plugged with tyloses. This is what makes things in the white oak family much more durable than things in the red oak family. But if you have tyloses that's plugging it, if I'm making a wine barrel, what do I want to make a wine barrel from? Do I want to make it from a red oak or do I want to make it from a white oak? Well, and same thing for building my ships. If I'm going to build a ship, I want to use a conifer or something from the white oak family because both of those are going to be non-porous. Things in the red oak family are going to be porous. So that's extra tidbit. So again, if I want to make a wine barrel, I'm going to use a white oak because I don't want it to leak through the barrel. Not all conducting elements transport water. So again, remember that all this white stuff is still alive and it's old vascular tissue. When it's produced, its original purpose is to carry dissolved minerals and water up to the canopy of the tree for the use in photosynthesis. So that doesn't always, that's not going to happen. The up and down part doesn't happen after the first year or so. As it says here, conifers may have 2 to 12 rings that may actually conduct water. So conifers are a little bit different. Most hardwoods only have 1 or 2 rings that are conducting up and down the tree. Now there can still be lateral movement inside of this tissue. Remember the ray cells that we spoke about earlier? Those ray cells that moved across those growth increments. If you remember, it crossed those annual lines. And by doing that, it can use those ray cells to move things throughout this symplasm. Again, sapwood contains many living parenchyma cells. Heartwood tissue is non-conducting. So once it changes color, this tissue does not conduct anything. There are no ray cells that cross into this area. If you looked at it under the microscope, there's actually a boundary layer that forms. As that boundary layer forms, these cells, there's a set of cells in there called intercellular material called cytocysts. These cytocysts explode and can fill the cell with phenols, which makes this heartwood slow to decay and inhibits fungi and other boring insects, things like that. So this non-living part contains no living cells, and sometimes it's darker in color. So we've talked about xylem tissues. So this is just moving things up the tree. Now we're going to talk about phloem. Remember, phloem just persists for the year that it's for the current growth year. After that, it's pushed out and is used in bark. Some of it may even be absorbed and reused. Some of the compounds can be absorbed and reused in other parts of the tree. So this is what's going to move the photosynthate around. Leaves produce sugars. These sugars are carbohydrates, and carbohydrates are what the tree uses for energy in one form or another. So energy is only manufactured in the chlorophyll, which primarily you're just going to find in the leaves. Some species you might say, well, Rob, what about hollies or crepe myrtles, things that have this sort of greenish tint to the bark? Yes, there are tissues even within all trees that can photosynthesize, like new spring shoots are going to be green, so that has chlorophyll in it. If it's green, it has chlorophyll and it's producing sugars. Those sugars are only moved downstream or locally right around where they are produced. So primarily movement is going to be from the leaves down the phloem tissue to where there's a demand for its use. So again, think of the xylem tissue can move moisture from the soil to sometimes hundreds of feet up into the canopy to be used in the process of photosynthesis. Phloem tissue, on the other hand, is going to move photosynthate downstream primarily. So it's primarily going to move from where it's manufactured down, and it gets consumed all along the way. So there's lots of tissue inside of a tree. Remember, all that living cellular tissue in the xylem has to be kept alive. All those parenchyma cells have to be sustained. All those roots have to be sustained, even though they're not making energy. They have to consume energy to grow. They have to consume energy to stay alive. So there's going to be a demand for energy throughout the canopy. So at the very top of the canopy, it's going to satisfy, say, the leaf itself. The leaf has cellular tissue in it that requires some energy to keep alive and doing its thing. The stem is going to consume some, the twig will consume some, the branch will consume some, and then the trunk itself will consume some, and then whatever's left over might wind up way down in the root system somewhere where it's going to be consumed. Again, think of, there's thousands and thousands of leaves on a tree, and trees produce enormous numbers of calories an hour. So there is lots of leftover. And we'll talk about that a little later. But this movement is also relatively slow and occurs across this pressure gradient. So it's going to be a higher concentration right by the leaf than it is much further down the canopy. Movement within phloem requires energy. Remember when we said that xylem tissue does not, those are dead cells, so it's not using any energy to move things around. These cells, however, these phloem, these sieve cells, are alive, and they basically have to make decisions. Maybe not decisions like we think of, there's not a consciousness, but it has to say, okay, I'm taking energy here. Does anyone have a demand for it? Yes, you have a demand. I'm going to push it through my cell wall into these companion cells. These companion cells will pull it out of this tissue and move it out to where it might need to be stored. So it is primarily composed of living cells. In the older exams, we drilled down and we discussed all these individual cell types. That's really not so important in the current edition. So that's why these have a line through them. So here we have xylem vessels and phloem vessels side by side so that you can kind of see what's going on. These tracheids, here's the actual vessel. This is going to be what carries everything up. Here it does show two-way movement through the phloem vessel, but again, this upward movement is going to be fairly local. If I'm a leaf on a stem and I have a bud here, I can produce energy and move it out to this bud. So it might move upward and outward a few inches, but it's not going to move upwards hundreds of feet. It won't move from one branch up into the canopy to be used at another branch. Why do you think that's important to understand? It becomes very important to understand when we deal with pruning, to really know and understand where energy is stored, where those starches are stored, and how they can move. You can't say, well, I'm going to cut all the top off the tree, and then these bottom branches are going to send energy up to help put on new foliage. That's not the way it works. It can't move that stuff up into the canopy. There's no ends to these things, so this is just this xylem tissue is just an open tube, one way only, and that's upward. The cells, you can sort of see these lines, so the cells are just stacked end on end. It's a fairly thick wall to make it durable enough to, when the tree's blowing in the wind, that they don't break and come apart. The phloem cells over here, we have the companion cells, the food storage areas. There is two-way movement. There's water and sugar. So, sugar by itself is a solid. So it has to be in solution. So there is some water that goes back down. These cells do have end walls. So these look like little barrels with holes in the end in order to allow that movement. So a little more about this phloem. So old non-functioning wood and its contributions. So here we have this phloem tissue. So the older the phloem tissue gets, these older cells, that remember, these are the newer cells right by the cambium. So this is the opposite of the xylem cells. So the newer stuff is going to be right here, and the older stuff is going to be towards the bark. Whereas in the xylem tissue, the newer stuff is going to be right here, and the older stuff is going to be towards the center. So this older phloem tissue that's out here is going to be absorbed and used in the cortex. So we'll talk about the axial movement of the transport system. And so we'll see there is longitudinal movement in the phloem and the xylem. Radial means extend across growth increments in what we know as rays. Rays are composed of parenchyma cells arranged in small layers. So we saw those little stripes. If you take a cross section of wood, you will see these cells that move, these lines that move across the annual increments. And this is different at every height in the tree. So these things are only a cell or two wide. So it's not like there's continuous ray cell all the way down the tree. So these ray cells, you could think of them more like a straw that's laid across the annual growth increments. And this straw goes all the way out to the phloem. So why is that important to understand? That it crosses the cambium into the phloem. Well, it's important because remember, starches can be stored in all of this living tissue that's not conducting things up and down the tree. So as there is a demand for nutrients, it can move stored nutrients out to that tissue, and it can either move up locally or it can move downstream for use in root development. Or if there's an attack on the tree or a damage to the outside of the tree and it needs to grow a new tissue and it needs extra energy to do that, it might move it from these ray cells out towards the phloem. So rays transport carbohydrates and other compounds into the sapwood as well as out of it. They store carbohydrates as starches. So as it pulls this leftover photosynthate from the phloem and it goes to store it, it's going to store it in a more stable carbohydrate form than it's actually you'll find in the phloem tissue. But it's still a carbohydrate. It will assist in restricting decay in wood tissue, and these ray cells are very important when it comes to compartmentalization of decay. But we'll show that photographically when we get to that section. They are living channels of cells through which water elements and carbohydrates may move laterally. So trees do a lot more than just produce sugars. They produce all kinds of compounds. As we know, a lot of medications come from trees. These are compounds that are naturally occurring inside of these organisms. So just keep in mind there that it can move more than just water and carbohydrates. It can move elements also. Now, bark. Bark's a pretty important thing. It's the outer covering of a tree's branches, the stems, and you might even see some bark on roots. It's composed of protective, non-functional phloem and corky tissue. The amount of corky material varies greatly by species. If you think about a bottle of wine, that cork that you see, the natural cork, the real stuff, not the synthetic, but the real stuff comes from these cork trees. It's a type of oak that has very thick bark, a very thick corky layer, and so they harvest that. That's what we use for our wine bottle corks. Compare that to, say, a crepe myrtle. Crepe myrtles have very little, if any, corky tissue. So you can see it varies greatly in thickness by species. These cell walls are impregnated with wax and oil. These naturally occurring waxes and oils can plug all the holes up and minimize the water loss as a result. bark can also moderate the temperature inside of the stem so that it doesn't freeze to death or boil off in direct sunlight. It provides defense against injury. It can be a physical barrier against, an animal running into the trunk, or physical defense against birds and mammals and stuff scratching around, climbing up and down the trees. So it does provide some of that. Some other parts of the tree that you might not often think of, or think that photosynthesis is a really complex chemical process. And if you've ever taken chemistry, you know that when you combine elements, just take, for example, H2O, hydrogen and oxygen. You have two hydrogens and a single oxygen. Well, what if I put in equal parts of both of those? I wouldn't have H2O2 because the natural formation is H2O, which means I'm going to give off. I'm going to have to liberate from the chain, from the reaction of that combination. The result's going to be water, H2O, and then there's going to be leftover oxygen, which is a gas, not a liquid. And so that gas is going to be liberated into the atmosphere after the combination. So there's lots of that stuff going on during photosynthesis. And in the combustion of carbohydrates, because remember, these cells have to stay alive. So they're going to take some of these starches and go through a process to change it into energy, adenosine triphosphate, and produce things, make things happen. So as it does that, it's going to do it in certain increments. It's chemistry. It's a chemistry laboratory of combining things and liberating things and absorbing things. And so we have these little things called lenticels. So these little specks that you see along this twig, these are little ports where these gases can go in and out. If I need more oxygen, I'm going to pull it out of the atmosphere to use it in my chemical reaction. If I'm producing carbohydrates, or if I'm producing carbon dioxide as a result of respiration, I need to be able to get rid of that carbon dioxide. So I'm going to push it back out of those lenticels. So lenticels are there to permit gas exchange. In some species, these can help you actually identify the species. Unfortunately, Kate's not here today to give this lecture on tree ID, but you will find it when you get into her section on ID. That there's lots of clues as to what species a tree might be. Think of something with a very, very large lenticel. Hopefully you can picture in your mind something like a cherry tree, a Yoshino cherry, if you're from sort of metro Atlanta, where you would have these trees in abundance. You see that silvery bark that goes around, and you see those big black spots along the branches. Those big black spots are actually lenticels. So those are big gas exchange ports. And then you can see something on an oak or a maple where these things are super tiny, like you see pictured. You guys are not asking any questions at all. So again, if you have a question about something, feel free to drop it in that Q&A. Now we're going to talk about stems. Stems are small. They provide support for, or twigs are small, and they provide support for leaves. So if you think about sort of this progression, you've got the leaf, you got the twig, and it gets bigger and bigger, it goes back down towards the trunk. So we have these small stems. So the leaf is on a stem, the stem is on a twig, the twig is on a branch, the branch is on a trunk. So that's sort of the progression of getting larger. We'll talk about here stem anatomy. So we have this is, you'll find this, you might even remember this from your 4th grade science class. You might have looked at it again in high school biology, and you probably looked at it again in college. So this thing has appeared to you several times if you've studied any biology, plant biology at all. So again, here's our terminal bud. This terminal bud is where the apical meristem is going to be. Normally the most active on each branch or twig. So if you think about, branches, you've got a primary branch growing straight up, and then you have some side branches. you're typically going to have this center one. It's going to grow more or have more extension than all the other branches around it. So this section is going to be the most active. We can see the lenticels, these little dots that are on here. And then we also know that these things get longer every year. So you have what's called shoot elongation that happens every spring. There's lots of chemistry that goes on out here in this terminal bud. There's an accumulation of a chemical called a gibberellin. That gibberellin accumulates all growing season, goes into dormancy, and then at spring that gibberellin is released, and you get this huge shoot growth, or this elongation of the stem. And then throughout the year, after that massive shoot growth in the spring, then you have shorter and shorter amounts of growth throughout the growing season, but you do have some incremental growth throughout the entire growing season, and then it stops, produces a new terminal bud. Once that new terminal bud is produced, things really slow down, and then it stops. So right here would have been the previous year's terminal bud. And so the distance from here to here is the length that was grown in 1 year's growing season. And so you can find these terminal bud scars. You'll see lots of scars on twigs, but look for the one scar that goes all the way around the twig. Those are going to be the ends, or the previous or the older terminal buds. So if I went down this stem, I might find another terminal bud scar down here. The distance between these is these ends are called nodes. And so here's a node. There's another node down here. There's another node up here that's going to be produced. And so in between the nodes is called internodal. It's the area between the nodes. The nodes themselves are where these scars occur. So you say, well, why is this so useful? Well, if I'm looking at a tree and I want to know if the tree is in decline, if it's in good health, I might go around and look or measure, take out my tape measure and check the internodal distance, and say this one is 4 inches and say this one is 3 inches. That tells me that the tree grew more this year than it did the previous year. So if I walk around the canopy, sample more than one area, I will find that in general, it grew more this year than the previous year. And I might think to myself, well, what happened? Did we get more rainfall this year? Did we fertilize in the spring? And this is a result of fertilization. Was the previous year really good? Did it store a lot of energy the previous year? And we got lots of rainfall to be able to utilize it for tip elongation this year. So these are just all diagnostic tools that you can use to see if a tree is becoming stronger or more vigorous, or if it's losing vitality. If these things were continually getting shorter and shorter, that tells you that the tree is running out of energy and not storing energy. And go to a construction site and you might see, it was doing 6 inches a year, and then all of a sudden it goes down to 4, and then the year after that it goes down to 3, and then 1 inch and 1 inch, and then a half an inch. And then 8 or 9 years later, it might pick back up again, and the internodal distance will return. So you can see the stress that construction caused to that tree by using the internodal distance. We also have these lateral buds. These are what might form leaves. Under it, you would have a flower bud. The previous year's leaves are going to produce a scar when they fall off. The scar pattern is also indicative of species. So every species has a fairly unique bud scar. So if you learned the sort of shape and appearance of these leaf scars, you could also use that for plant ID purposes. Off of these shoots, like this one had this little side shoot, maybe like where this leaf bud here is this year. The previous year, it might have looked like this. And by the end of the season, it grew to be a little twig and produced another little meristem out here at the tip. But this one's still going to be this will be a terminal bud, but this one's still going to be the apical bud out here. So even here, I can have a node. This is how far that node grew. So this one next year might be out here, and then there'll be a node there. So hopefully you can see this in 3 dimensions over time. So a lot of these buds, here's another lateral bud, these are not always used. If the tree is producing enough energy, it produces, it puts out a set of leaves in the springtime. If everything's happy, then great. But if we have caterpillars or something that come along and chew these leaves off and it needs to put out a second set of leaves, it has a backup set. Many species even have a tertiary set or a third set of leaves that it could push out in a single growing season. So under normal healthy conditions, these secondary and tertiary buds are never used. And as secondary growth occurs, these buds get buried in the vascular tissue over time. However, when the tree something happens to it, it's stressed out, it's over pruned, it's exposed to more sunlight, there'll be some stimulus. So this stimulus will produce adventitious growth. So those old covered up bud scars become what are referred to as meristematic points. So if you've ever pulled the bark off of a trunk and run your hand along the bark, remember I talked about it being sort of slick, run your hand across that bark, and every once in a while you'll feel these very sharp points. Those are meristematic points, and they can be turned on from a stimulus trigger and will result in this adventitious bud development. So typically these buds are suppressed until they're called upon. Once they pop out, they can be referred to as epicormic shoots. So it looks like we're about at 8:59. So I'm going to give you guys about a 5-minute bathroom break. Is that good? You there, Brittany?
Yes, sorry, I forgot to turn my mic on. Yeah, definitely. Everybody, if you could be back at 10:05, we'll get started again.
Thanks. See
Thanks,
you guys
guys.
soon. All right. So we should be back now.
Yeah, it looks like everybody's making their way back. We're down to just a few seconds. So before we get started, you do have a question in the Q&A that came up during the break. I think this is regarding the epicormic sprouts. Jeremy wants to know, are they harmful or just a sign of stress?
So the tree produces McCormick growth or the sucker growth in response to something. The response could be a positive response or a negative response. However, the tree requires or can utilize more leaves. So sucker growth is actually good because it helps to produce more photosynthate and the more chemistry for defensive chemicals and things like that. So we always think of the leaves that are produced as being a positive thing. Why they are produced could be a response to ill health, but the actual leaves themselves are always good. Hopefully that answers the question. And we will talk about sucker growth more as we get into pruning and pruning response. But just know for now that these are a response to a stimulus. All right, so we're going to get going back with branches, where we left off. How exciting. So branches, first thing that you have to understand is that branches are autonomous. Think of it as another tree. Energy and growth is primarily occurring out at the tips, and it moves this energy inward down the branch itself. The branch does not really receive anything from the trunk. So there's not going to be energy stored in the trunk that's going to move out into the branch to keep the branch alive. There's no welfare system in trees. There's no method to send money or send energy out to someone that's not producing enough at the moment so that they can reach out into the sunlight and become a producer again. They really can't. This isn't a concept for trees. Trees just move energy inwards towards the trunk and down towards the root system. So whatever required to keep that branch alive has to be manufactured by that branch. So again, if you think about pruning, that's why pruning is not so great, because you're pruning off its ability to keep itself alive. There's a unique attachment from the branch to the actual trunk of the tree. And we'll take a look at this application during pruning. Branches are strongly attached to wood and bark below the branch, but weakly attached above the branch. So it's a sort of vigorous growth at the bottom. Think underneath the tree, there's the branches being compressed against the trunk, but due to gravity, it's being pulled down. Wood is very strong in tension strength or the pulling, so it doesn't require as much on top of the branch to keep the branch in place as it does under the branch to help support that branch and the branch collar. But the actual form of attachment, just think of it as being weaker above and more strong below. Branch morphology. Remember, morphology is the appearance that is taken. So layers of tissue produced annually at the junction of the branch and the stem create the branch collar, or that bulge or that shoulder. And we'll see that in the next slide. Where the branch and the trunk expand against each other, the bark is pushed up, creating a branch bark ridge. When the bark in a crotch is surrounded by wood, it can become included. So the picture that we have shown here is included. This is included bark. So these two are kind of growing independent of each other. There's not really a parent-child relationship here. They are co-dominant in nature. So you'll also notice that there's not really a branch collar seen. Again, you haven't seen one yet, but here's what a branch collar looks like. This is that swollen tissue. Here we have a much smaller branch, a larger trunk, so you can see the parent and the child relationship here. This tissue growing up the trunk is wrapping around the tissue being produced by the branch. So the branch is going to grow in lateral growth every year. The trunk's going to grow in lateral growth every year. They each have their own vascular systems, and so they sort of weave in and out of each other and form this sort of doubled up area of tissue. So this branch collar shown here is this side of the collar is a combination of branch and trunk tissue. If I look out here, this is just trunk tissue. This is that branch bark ridge that we talked about. So this is where the wood from the branch junction is pushing up against the material of the trunk. So everything on this side of the branch bark ridge is just trunk material. The stuff on this side is going to be trunk and stem material. Remember, this grew in diameter every year, and this grew in diameter every year. So if we went back in time to see when this branch bark ridge started, we see it somewhere right in here. Might be helpful to use pen here. So if I went straight up the tree from that point, straight down the tree from this point, this branch started about right here and grew out in this direction. So this would have been the diameter of the trunk the year that the branch was sprouted off. And so from this point, the trunk produced some tissue, the branch produced some tissue, they crush up against each other, and you can follow that line all the way out to today, where we see it up here. So hopefully that will give you some 3 dimensions to this, to what's happening inside of here. And why is this important to understand? Well, it's really important when it comes to pruning, because you don't want to prune any trunk material. You want to do no trunk damage at all. Remember, damage is permanent. So if I prune out here, this is just stem wood. Everything on this side is just stem wood. There is no trunk wood. So for those people that like to cut the branches off like this, as Dr. Kim Coder likes to say, to slick those branches off, that's going to cut into not only the stem wood, but also the trunk wood, because remember that stem and trunk wood are both in here. So decay has to happen, or damage control has to happen in 2 different places. It has to happen for the trunk and for the branch. Out here, if I cut it, the branch is the only thing that's going to respond. The trunk won't know that it ever happened. So then you'll get some callus growth after this. Now let's go back to this included bark business. So as each of these grow in diameter, they touch each other. They begin to crush each other. So I'm sure you've all seen a Bradford pear that's just sort of fallen apart. These Bradford pears fall apart because they are all cloned. Cloning meaning that there's one parent Bradford pear out there. And this Bradford pear was produced over and over and over again, so that what we see in our environment is a very predictable tree with the same shape, same growth rate, flowers at the same time, all these sort of things that architects love to have. However, the drawback to it is that they are structurally a disaster area because of all of this codominance that occurs. So as each of these grows in diameter, they put more and more pressure in this area of inclusion. And on Bradford pears, when they get to be about 10 or 12 years of age, or when they get to be about 14 or 15 inches in diameter, they start breaking apart. So everybody says, well, they're very short-lived. Well, they don't have to be. If you pruned out a lot of these defects when you first planted them, they would never have been a problem. Pear trees can live well over 100 years. So there's no reason a Bradford shouldn't be able to live over 100 years. Also, you just have to prune to eliminate the occurrence of this included bark. And we are going to beat this included bark thing to death before the end of this 7 weeks. So let's also take a look at the leaves out here. Leaves produce food for the tree. Leaves have cells with chloroplasts that contain chlorophyll, which is the green pigment. Chlorophyll is the primary leaf pigment that absorbs sunlight. The energy of the sunlight is collected in the chloroplasts, and the light energy is converted to chemical energy, and we get carbohydrates as a result of photosynthesis. this little diagram showing us a few things. We have the cuticle. This is the waxy covering on the outside. We have a vascular bundle. So in leaves, remember, most leaves are disposable. They're only going to persist for 1 year. Some trees, they might persist for 2 or 3 years. You know, they have a limited lifespan. So there's no need to go to this extra layer of complexity to produce annual growth rings inside of a leaf. So the leaf's just going to be disposed of. I just need a temporary solution to get things, to get compounds out to the leaf to be manufactured, and then I need to get the photosynthate moved out of the leaf to where it can be used elsewhere. So it does this by forming these vascular bundles, which is a combination of xylem and phloem tissue together. We have parenchyma cells. Remember, these are the living cells that do a bunch of work for us. They store energy, they help to move it around, all that sort of stuff. And there's lots of chemistry going on inside of a leaf, obviously. And so that leaf needs to be able to move gases in and out. It does that through these stomata. And the stomata size, the aperture is produced by reaction from the guard cells. So these are the guard cells on either side of this opening. We'll look at those a little closer. Leaves are also where transpiration occurs. Transpiration is the liberation of moisture, or this water. It's the loss of water vapor from the leaves, which cools the leaf and draws water up through the xylem. So as it's liberated, it produces sort of a negative pressure or a vacuum. And remember that the xylem tissue is a continuous straw of dead tissue. And so as it's liberated off the top, it's able to pull in from the root systems, and this constant movement of water throughout the canopy is had. Leaf blades provide a large surface area for the absorption of sunlight and carbon dioxide necessary for photosynthesis. Leaves are also very thin. They're just a few cells thick, so that no cell is very far from the leaf surface, optimizing its appearance to the sun. The outer surface is covered in that waxy layer called the cuticle. The cuticle minimizes desiccation of the leaf. Desiccation, again, is the loss of moisture. You don't want your skin to dry out, so you might put on sunblock or some sort of moisturizer on your skin to help hold in the naturally occurring moisture produced by your skin. Same sort of deal with the leaf here. It's covering, it's coating to help protect it from sunlight. The thickness of this cuticle is highly variable depending on species. Something like a magnolia is going to have a very thick leaf, very durable leaf. A live oak is going to have a very durable leaf. Holly's very durable leaf. But then you take a look at understory trees. Things like our dogwoods have very thin leaves, a very thin cuticle layer. A lot of the shrubs that you find out in the forest have very thin leaves, like azaleas and stuff like that. And so because they're always covered in a canopy, they never had to produce extra waxy coating, extra cuticle. Small openings in the leaf, the stomata, control the loss of water vapor and exchange of gases. Guard cells regulate the size of that opening. Leaves have a network of conducting tissues that include the veins or vascular bundles. These vascular bundles are, again, composed of both the phloem and the xylem tissue and are there for transport. Here's a cross section of a leaf. Here's an actual photograph of the bottom side of the leaf, and it's done under a microscope so that you can see the cells. These little black dots are the nucleus of each cell. Here are the guard cells around this is the gas exchange port. Notice on the bottom of the leaf, it's a little easier to see in this diagram that this bottom of this is pretty spongy. It's the spongy mesophyll and with lots of airspace. So remember, all this chemistry is going on. Gases are liberated, gases are used. And so anytime you have gas exchange, gases can expand and shrink easily. Now, if you remember anything about Boyle's law or the study of gases, you know that a gas can occupy equally and uniformly any space that it's put into. So if you put oxygen into a room, then oxygen is going to go all over the place and expand. The hotter it is, the more it wants to expand. The colder it is, the less it wants to expand. So you have to account for this expansion and this shrinkage somehow, and it does that in this spongy area. So it can move, it can swell, it can shrink easily to accommodate all these gases being liberated or taken in. Leaves or we refer to them as either deciduous or evergreen. So notice we have both of these are in the oak. Well, I don't know that this one's necessarily an oak, but it could be. And this has dropped its leaves in the winter. In the background, you might see some conifers in the background, or you might see some evergreen trees. Our live oaks, for those that don't know, I'm in central Texas right now. I'm just outside of Waco. I'm not in Georgia. So our trees here, our live oaks, are very common, and they are evergreen, meaning they always have a set of leaves on producing photosynthesis throughout the year. They do not go into total leaf drop. That does not mean these leaves last forever. They might only last for a year or two, 2 to 3 years, 2 to 4 years, depending on the species. Magnolias are the same. They're going to have those green leaves that are going to hang around for a year or so after the new ones are produced in the spring. Some of the older ones are dropped off. So you have this sort of natural cycle of leaf retention, but it causes us to call them evergreen. When the leaf falls off, you don't want it leaves that scar. Remember that vascular bundle scar, that leaf scar that we saw on the classic stem diagram. The tree doesn't want to lose a bunch of moisture. It's just like if you have a cut, you don't want to like bleed out. You want that cut to close up. So the leaf has this special area called the abscission zone. The abscission zone morphs over the fall to the point where it can separate the leaf and the leaf falls off, and the remaining section on the stem is not going to be open to the atmosphere for fungi to get in or for water to get out. This area is a protective area against desiccation and pathogen entry. Leaves are shed in a response. So when fall happens, this abscission zone, once it's triggered, it wants to get as much of those expensive compounds that it spent a long time manufacturing and accumulating. It wants to reuse those, wants to recycle them. So it's going to pull them out of the leaf. As it does that, you're going to see different colors produced in the leaves. So as it pulls out, certain compounds, what you're going to be left with causes the leaf to turn red or to turn yellow or to turn purple. And so you get all these cool colors in the fall as a result of how or what speed it pulls those compounds out of the leaf. Once all those useful things are out of the leaf will begin to shrivel up and then it falls off. Well, what triggers all this to happen is a response to how much sunlight there is, how much temperature there is, and or how much soil moisture or rainfall is there. Environmental changes cause the cellulars at the cellular level to change. Growth regulators form the abscission zone at the base of the leaf stalk or the petiole. You might have read about a product called abscisic acid that trees produce, and it was originally thought that abscisic acid acted on the abscission zone to force the leaf to fall off. However, studies now show us that that's no longer true. So, but we still call it abscisic acid. So just sort of wanted you to know where that came from. Short, sunny days and cold nights are going to enhance the accumulation of sugars and trigger a decrease in chlorophyll production. And then you're going to get all these cool colors. Here's sort of an extra slide about tree colors or leaf colors and where they come from and why they're produced and where they're produced. You can look at that at your leisure. So now we're going to go from leaves to the root system. Roots, this is where moisture and oxygen are I'm sorry, roots grow in response to the environment that they're in. Roots can only grow where there's enough moisture or sufficient moisture and oxygen. Remember, roots are not producing energy. So if you're not producing energy, you are consuming energy. And if you're consuming energy, you require oxygen to break down the carbohydrate to be released for energy, and you give off carbon dioxide. Does that sound familiar? Yeah, well, it should, because that's what animals do. It's what people do. We breathe in oxygen, we give off carbon dioxide. So there has to be enough oxygen in the soil for that process to occur in the root system. And you cannot have the soil so wet that there's no airspace, because without airspace, you're not going to have oxygen available either. There's 4 primary functions to the root system. We have anchorage. It's going to hold the tree from blowing over. And you're going to have absorption, storage, and conduction. And you could think about anchorage in 2 formats. You could think about anchorage as a compression, and you can think about anchorage as tension strength. So as a tree stands up and it moves to this side, if the wind is blowing in this direction, it's pushing the tree over here, meaning that it's going to compress on this side and it's going to pull on this side. The combination of compressive tissue and tension tissue is what holds the tree straight up and down, or allows it to endure this movement to dissipate those forces into the soil. So anchorage is not only just tension strength, it's also compression strength. Roots absorb stuff out of the soil. They can store moisture, store water. They can also store extra carbohydrates. Remember, all those leaves are producing photosynthate that's moving downstream, and all the leftover stuff has to wind up at the root system to help it out. Yes, ma'am. I see you popping up over there.
Yeah, you've got a question here in the Q&A. The question is, are there theories as to why the leaves require CO2, but the roots need O2?
Yeah, so carbon dioxide is used in photosynthesis. So it requires carbon dioxide to be brought in. That's part of the chemical combinations that occur during photosynthesis in order to produce sugars. If you think about what sugars are, they are carbon compounds. So the easiest or most abundant form of this carbon compound found in the atmosphere is carbon dioxide. So it's pulled in to use in photosynthesis, but to use energy, you have to break down that same carbon molecule. So when you break it down, what are you going to have left over? You're going to have carbon dioxide left over, so it liberates it back to the atmosphere. But that breakdown has to be in the presence of oxygen. So just like in our bodies, we have to breathe in oxygen, which is going to go through, we could get into the Krebs cycle, and you could get into ATP or adenosine triphosphate. We could get into the chemistry of it, but you don't really need to go into that detail for the test. But when you're consuming energy, you give off carbon dioxide. If you're a tree, if you're producing energy, you're going to take in carbon dioxide where energy production occurs. Where energy liberation occurs, that's where you give off carbon dioxide. Does that answer the question? I hope that answers it for you.
He said yes, thank you for the explanation.
Okay, good. And so we're also going to, so not only are we going to absorb things from the soil, from the very fine parts of the root system, we're also going to conduct it into the xylem tissue and eventually push it up into the top of the canopy where it can be used in photosynthesis. So there's 2 types of roots that we'll talk about. The main are the large woody roots. These anchor, store, and conduct. And then there's very small, fine roots called absorbing roots. These are fibrous. This is primary tissue that grow at the ends of and along the main woody roots. Absorption roots are very, very small diameter roots. We'll see some micrographs of it in a few minutes. Absorbing roots have epidermal cells that may be modified into root hairs. And again, we'll see a picture of that. Root hairs aid in the uptake of water and minerals. If you think about an ice cube, if you have one large ice cube in your glass, you might be able to keep something cold for quite some time, but it's not going to make it cold in a hurry. If you have crushed ice in a glass and you add something to it, it's going to make whatever's in the glass very cold very quickly, but the ice isn't going to last very long because the ice has lots of surface area. So same sort of thing with the root. If you have a cell that produces a hair that comes up out of the cell, you're increasing the surface area of the cell, making it more absorptive. So root hairs, because they're so small, this is what pulls in or sucks the water, exploits the soil for the water and the minerals. Root tips contain a meristematic zone, as with a shoot tip, where the cells divide and elongate. We saw those in a previous slide. Most absorbing roots are found in the upper 12 inches of the soil. 98% of the root system is going to be in the top 18 inches or so. The majority of those are going to be found in the top 4 inches because that's where the air is going to be. So whenever you see these sorts of statements, most absorbing roots are in the top 12 inches of the soil, this is what you need to know for your exam. However, keep in mind that everywhere in the world is going to be a little different. Even within the United States, you can go up to places with glacial till soils where fibrous roots might be found 15, 20 feet deep, or you might go to highly compacted areas or high clay areas where it might be only 6 or 12 inches, or into the rainforest where it might only be 2 or 3 inches of thickness. So this is a variable statement. And so this test is written again for a global environment, so a lot of things are averaged, become a little generic, but the takeaway here is that absorbing roots are found in this close to the soil surface where there's oxygen and sufficient moisture. There are lateral and sinker roots. Horizontal or lateral roots are usually near the soil surface. Some trees form sinker roots, which grow vertically downward off of the lateral roots and provide anchorage and increasing the depth of soil exploited for the root system. So here's sort of an example of a sinker root where the root goes out and then another one sort of goes down. Again, the root still has to have enough oxygen. So if it's not, it'll only go as deep as the oxygen gradient allows. So tap roots are sort of a juvenile function, especially in the Piedmont region of Georgia. When we think about tap roots, it's really just a juvenile feature of any species of tree. Most species have a taproot at some point in their life, and what that taproot wants to do, let's take an acorn, for example. You have an acorn, the first thing it wants to do is it wants to put out a root and a couple of leaves at the same time. And inside of that acorn, there's lots of energy stored up. And so that energy is being pumped out, some to the roots, some to that stem, so that it begins growth. What that taproot does is, think about it as a sensor. It wants to go and find out how deep is my soil. And so it continues to grow, and it has this little meristem at the tip. Remember that, we talked about all the chemistry at the meristem, and we talked about the production of gibberellins and the storage of gibberellins right behind there. So this taproot is going to grow down until it hits about an 8% oxygen gradient. And so once it hits 8%, that's a little less than what it needs to stay alive. And so it's being falsely grown that deep because it's being provided energy from the acorn. But at some point, the acorn runs out of energy and it can't sustain that root at that depth. And so that little root cap dies. When that little root cap dies, it signals, or it's no longer the apical meristem. And so it's no longer in charge, and the lateral shoots can begin to come off of that main downward root. And again, that downward root could be 24 inches deep. It could be 12 inches deep. It's wherever it hits that 8% gradient. Very few mature trees actually retain a taproot. Everybody always asks, well, are pine trees different from hardwoods in their taproots? In the Piedmont region of Georgia, the answer is no. Both are right on the surface. You might have seen trees fall over and you see that big disc of dirt that hangs out of the ground. That's the compression system. That's what forms off of the root crown. And that compression system is what bore the weight of the tree onto the soil. So if you've ever seen pine trees fall over, you see that same disc of dirt that sticks up. So they behave very similar to hardwoods, especially in the Piedmont region of Georgia. They're really not going to have a taproot that's any deeper than the taproot would have been on the oak tree. So the area where the roots join the main stem is known as the root crown. So this whole area is sort of the root crown. Roots spread out from the root crown and decrease in diameter. So you have roots that come out like this. They're very large in diameter. These are first order primary roots that you see coming off of the trunk here. The number and the distribution of these around the tree is genetically predetermined, and it can vary even within the species. You might see a water oak that has 4 of these, or you might see a water oak that has 35 of these first order primary roots. Generally speaking, the more first order primary roots the tree has, the healthier it's going to be, the more stable it's going to be, the longer lived it's going to be in general. So that disc of dirt that comes up and falls over in a storm, those are the large diameter roots. At some point away from the tree, they go from being large diameter roots to being about the size of a silver dollar over a period of about 12 to 18 inches. That taper becomes the weakest point in the system. So that area is also known as the zone of rapid taper. So when you have a force that's blowing and the mechanical ability of the root system to hold this tree up finally fails, the whole tree is going to pick up and fall over. If this is the compression plate, the whole tree is going to pick up and fall over, and it's going to break here at the zone of rapid taper. Roots may extend laterally for long distances depending on the tree and the soil conditions. For trees grown in the open, roots can extend 2 to 3 times the radius of the crown in all directions. They can extend, if roots are successful, they can extend up to 10 times the height of the tree. So root systems can be quite pervasive given the right soil conditions. Root extent and direction is a function of the environment rather than genetics. It's like, okay, where is it going to be successful? This root's got plenty of soil moisture. It's finding lots of elements. The tree's going to spend a little more resources on its growth out into the world than I am on something that's constantly flooded or is constantly dry or barren. So give a house foundation, for example. A root's not going to want to grow under a poured foundation because there's no air, there's no moisture below a foundation. So it's going to run into the foundation hit, and then it might run down the side of the foundation, but it's not going to want to grow under it because it won't be successful. If you're on a slope growing down towards a water source, you might be very successful because you're finding an increasing moisture gradient as you approach the streams. And so you become very successful. There's lots of detritus on the ground, lots of decay, and you're finding minerals and nutrients to be used in photosynthesis. So your root might become quite long compared to one that's growing towards a compacted area. The trees can only absorb so much water and only so many minerals out of the soil on their own. They have a relationship with a fungus, and that relationship is called mycorrhizal. And so we have these mycorrhizae. These mycorrhizae are it's a fungus that grows or has a symbiotic relationship with the root. So it literally means root fungus. So you've got sort of this combination, but they are both benefiting. The root is being benefited from the fungus, and the fungus is gaining some benefit from the root. So the fungus might be getting in some carbohydrates to keep itself alive and keep it growing, and the root in turn is providing something like phosphorus to the tree. Phosphate is a very large molecule, and root hairs have trouble moving that molecule across the membrane into the system, but mycorrhizae can do it quite efficiently. If we look back 165 million years in time, we'll find that trees themselves did not have roots of their own at first. Their root system was actually just the fungal system. So you had sort of this tree taking advantage of the fungus, and now it's kind of the other way around. But fungi derive nourishment from the roots of the tree, and fungi aid the root in the absorption of water and essential elements by increasing the surface area. So here are two pictures. Notice these little dots, those are the nuclei of the cells. And so this is one cell. Out of that one cell, you get this little hair. So you can imagine that this is underneath a microscope to be able to see this hair. Well, this is under about the same amount of magnification. You can see how much larger this organism is than this little hair. This is a mycorrhizal organism. Mycorrhizae come in two forms that I don't think you need to know for the exam, but there's endomycorrhizae, which grows inside of the cell, and ectomycorrhizae, which grow on the outside of the cell. This is an ectomycorrhizal organism known as Pithylicis tinctoris. Off of these will grow this little white mycelial growth. So if you've ever been out in the forest, you pull back the leaves and you see this white stringy stuff in there that's coming from the mycorrhizae. They can extend or increase the size of the root system tremendously and exploit the soil as a result and help the tree along. So we'll talk a little about physiology now. Photosynthesis is putting together with light. It's a process by which green plants use light energy to build carbon molecules that make carbohydrates. Raw materials needed are carbon dioxide and water. Carbohydrates, this is the chemical energy used for growth and development or stored as a starch for later use. Oxygen is released through the stomata as a byproduct of photosynthesis. So again, as I'm consuming this CO2, I'm using the carbon part at the moment, and I don't need a whole heck of a lot of oxygen, so I'm going to get rid of the extra oxygen. Here's sort of an oversimplified look at photosynthesis. So it's going to collect everything it needs from the grocery store down here. It's going to travel along the interstate down to the highways, the local roads, till it gets to the leaves or the kitchen. And the kitchen's going to convert those things into something that can be eaten or consumed. And so that's the process of photosynthesis. And that's, as a result, you have the food that is used to keep everything else alive. You have an input of carbon dioxide, an input of water, and an input of sunlight. And as a result, you have the chemical reaction. Carbohydrates are produced during photosynthesis. The building blocks for many other compounds required by the plant is also produced here in photosynthesis, not just carbohydrates. There's lots of other things produced. Photosynthesis can produce proteins, starches, fat, growth regulators, amino acids, and other important compounds. Now, can you think of a tree fat? Can you think of a fat tree? I'll give you an example. An avocado is the most obvious. So if you cut into the avocado fruit, all of that stuff surrounding the center is a fat produced by a tree. So trees can produce fat. They can also produce oils, and oils can be thought of as a fat. Respiration. So we talked about the production of energy through photosynthesis. Now we're going to talk about the consumption of energy through the process of respiration. It's a process by which carbohydrates, originally stored as a starch, are now going to be used by the tree to stay alive, to keep those cells satiated. Carbohydrates are chains of carbon, hydrogen, and oxygen molecules chemically bonded together. When these bonds are broken off, energy is released and carbon dioxide and water are given off. Trees use released energy for all biological functions. Any cell that's alive is consuming energy and is therefore respiring. It's a constant process of converting food into energy. Important that photosynthesis or food production exceeds respiration. Why do you think this is important? Well, you have to have more coming in than you have going out. If you think about a triathlete, he or she's expending an enormous amount of energy, and it would be impossible to really sustain yourself in that level of aerobics before you go into a state called anaerobic. And anaerobic environment is one where you're consuming more energy than you can get a supply for. So if a tree does that long term, the tree dies. When respiration takes place in the absence of photosynthesis, the tree must rely on stored energy reserves. Think about, how does an oak tree stay alive in the, how does a red oak or a white oak stay alive in the winter? It has no leaves. It's not producing energy. But recall that it stores energy in all of that symplasm that it has, all that symplastic tissue that's part of the trunk, that old xylem tissue, all those parenchyma cells and those ray cells. This is when they go into heavy duty action to keep all the cells fed throughout the dormant period. So the trees never actually stop. They don't die. They stay alive all winter, but it's through these stored reserves. So over a long period of time, this usage of stored energy reserves will cause the tree to run out of energy and die. Again, if it's using more than it's storing, so eventually that leads to death. So oxygen is required for normal aerobic respiration. Trees cannot respire in an anaerobic condition for very long. Or in other words, the absence of oxygen. Think about flooded conditions or severe soil compaction. When oxygen is scarce, respiration uses food at a much higher than normal rate. Kind of like panic. If you're underwater and you're breathing on your scuba gear and something freaks you out, you're going to start breathing faster and faster as a response to that stress. Even though you know that you have a limited air supply on your back, you're still going to consume oxygen at a higher rate because you're more excited. So if you do this, if you have this anaerobic condition for long enough, it does result in root death. Now, a lot of this fiber tissue can die and be regrown several times in a season. On a wholesale effect, once you really start to kill off the main transport roots, those larger diameter roots, then you're going to run into long-term issues. Transpiration. Transpirational water loss in the form of water vapor is also affected by cuticle thickness, the presence of hair on leaf surfaces, number and location of stomata. So again, transpiration is the movement or the pull of water through the xylem tissue. Hot, dry conditions, you want a thick cuticle, small leaves, and sunken stomata. Stomates usually open during the day and close at night to conserve moisture in the tree and to hold that column of water in the dead tissue of the xylem. Antitranspirants, these are sprays that can be used to close the stomates on purpose. If you know you're going to stress a plant out, say you're going to transplant it from one side of your college campus to another side of the college campus. So you're going to dig it on Monday and you might not plant it until Wednesday or Friday. An antitranspirant might be used to keep the tree from losing too much soil moisture, too much tissue moisture. And so that when you put it in the ground, most of these compounds wear off after a few days and the tree starts doing its business again. So that's an example of where antitranspirants can be used. However, they can be dangerous if it's a drought condition or it's super hot. It could make problems worse and it could actually kill the tree. So you really have to know what you're doing to use antitranspirants. They can reduce evaporative cooling, uptake of carbon dioxide, and photosynthesis. So water is essential for all living cells. Photosynthesis, it's a building block in photosynthesis. Remember, you have to have these hydrogens and carbons, and so where does it get the hydrogen from? It gets it from the water cell. It's used in the actual process of photosynthesis. It's used to hold the leaves out nice and tight. So if there's not enough soil moisture, the leaves wilt. So there's a wilted state and a turgid state. Most leaves are going to be in the turgid state when there's plenty of soil moisture. It's necessary for the transport of essential elements. So the tree's not going to pull dry elements out of the soil and be able to move them up a dead tube in that dry state. So they have to be dissolved in the water. So water is essential for that solution to occur. Water is absorbed with minerals and elements from the soil by the roots. It's used for growth and metabolism. Most water is lost through transpiration. So remember those stomates on the bottom side of the leaves and all those lenticels, they're all going to be liberating some vapor into the atmosphere, and that's where most of the water is lost. So if you thought about just sort of a fountain that's pulling water up out of the source and spurting it out the top, and it falls back down to the ground, and it's used, and just the cycle goes over and over again. That's sort of what's happening here. That needs to be nice and continuous. If you shut the pump off, it's harder to get the thing going again up a tall fountain. So you have to have a very strong pump to get it going again, but once it's going, it's fairly economical to maintain. So that water loss, that constant evaporation or that constant liberation from the leaf is what creates the transpirational pull, or that vacuum that helps to stand that water column up regardless of the tree height. Xylem is a continuous column of water. Water enters the young roots or the mycorrhizal roots by a process called osmosis. Osmosis is the movement of water through a membrane from a region of high water potential to a region of low water potential. Pure water has the highest potential. Pure water has a psi of 0. All else is a negative number, such as a psi of -1. Water with minerals or sugars lowers the potential. So the more there is in solution, the more negative this number becomes. Water with minerals or sugars has a lower potential. Water normally moves into roots where the water potential is lower than in the surrounding soil. If water potential is lower in the soil than in the roots, water will move out of the roots into the soil. This might happen in a couple of conditions. If you have a severe enough drought, your plants wilt because the soil actually begins to suck the moisture out of the plant. Also, if you have lots of salts along your roadways, the same sort of thing can happen. If they're using salt for de-icing, and then the snow removal company comes and moves the snow off of the road, blows it onto the roadside, it gets on top of the tree roots, and the salts get into the soil, and those salts can pull moisture out of the tree as well through that osmotic process. We think about energy being produced, stored, and consumed. So we have this source and sink analogy here. Photosynthate moves through the phloem from a source, that would be the leaves, to a sink, that would be anything basically that's not a leaf producing photosynthesis. Leaves are the source of the photosynthate. Almost all parts are sinks at one time or another. Remember that the wood is not alive, so it's not producing anymore. There are no stored starches in that area. Most photosynthate is utilized or stored in proximity to where it's manufactured. So at the leaves is going to be where the highest use of that leaf's sugars are going to be. Excuse me. And so it's going, whatever's left over then moves down. So the concentration away from the leaf where it was produced downstream becomes less and less significant. But there's thousands of leaves, so there's still a mass movement of photosynthate down the branch. Here's an example. Here's our leaf. There's that companion cell that's within that vascular bundle. That thing's going to move it along. It's going to dump it over here into the phloem, and the phloem can send it downstream over here to be used. So it's going to pull out some of the carbohydrates, and there's going to be some water that is remaining, so it can actually contribute that water source back to the xylem tissue, as well as other waters that are coming up from the base. So this contribution also helps to stand that water column, help keep that water column going. But this is where this companion cell is going to say, well, my cell needs energy, so it's going to take energy out of the phloem across this membrane into a, say, a root cell that's going to be a sink. Movement of water in the xylem and photosynthate in the phloem are examples of longitudinal or axial transport. Radial transport is the horizontal movement of water or nutrients within the tree between cells of different ages, primarily through those ray cells. The result of the interaction between genetic potential and environmental conditions, such as urban versus natural environments. Remember, we are arborists, not foresters. You may also be a forester, but here we're talking about arboriculture, which is a study of trees as individuals, more likely seen in an urban environment rather than in the forest. So things that are affected would be the size of the tree, the fall color, the form. So we have some genetic potential to get quite tall, provided we have enough sunlight or we're not covered up by taller trees already. The fall color, you know, you've all seen these autumn blazes. These are chosen for their genetic potential to be a very bright fall color. The form that they take, are they going to be upright and cylindrical, or are they going to be wide and spreading? All that will come about as a result of its genetics. Plant systems respond to environmental stimuli, such as light, gravity, and temperature. There's other stimuli, but these are the primaries. Processes in trees are controlled in part by plant growth regulators or hormones, plant hormones, naturally occurring compounds that act in very small quantities to regulate things like cell division, elongation, flowering, ripening. These are the communication tools that the tree has to trigger responses. The primary plant hormones that we'll talk about and you'll be tested over are auxins, gibberellins, cytokinins, ethylene, and abscisic acid. Remember, we talked about where this name came from a little while ago. Auxins are produced primarily in the shoot tips, so you'll find them out at the ends of the roots. You'll find it out at the ends of the canopy. Primarily in the canopy, you're going to find these auxins are produced. Auxins are limiting functions. It tells the rest of the twig or branch, hey, I'm in charge. I get to grow more than you do. The auxins can be found in synthetic forms to to do things like root cutting or twig cuttings to produce roots faster, things like that. Auxins can also be involved in tropisms, or the orientation of the direction of growth in response to external stimuli. Geotropism is the upward orientation of the stem growth and downward orientation of root growth. Phototropism means that it's going to grow towards the light. You see these trees that grow out at an angle over lakes because the sunlight's always going to be over the lake. Whereas in the forest, the sunlight's always going to be above, so the trees are going to want to grow straight up. Cytokinins are produced in the roots and are instrumental in shoot initiation and growth. Auxin and cytokinin exist in a delicate balance in plants, regulating shoot and root growth. So the relative abundance of these two compounds change cyclically throughout the year, or phenologically throughout the year, to make different things happen. Apical dominance. So the growth regulators present in the terminal buds, again, inhibits the growth downstream. Strong dominance is confined primarily to the current season shoot, after which the lateral buds begin to grow. So that first year, you've got very strong dominance, and then the next year, you still have strong dominance, but a little more dominance by the offshoots to the main. If lateral shoots outgrow the original terminal shoots year after year, a round-headed or a decurrent tree will result, such as oaks and elms. Excurrent trees have strong apical dominance, resulting in upright trees with strong central leaders, things like sweet gums, poplars, and most conifers. All trees start out with kind of an excurrent trait. All trees want to get up as fast as they can, find the sunlight as fast as they can. So that's typically going to be on a single main leader. However, as they mature, that might change. Here's an example of X current. So you have a strong central leader and things coming out off of the main leader. Here's a D current leader. So you see how this one's much more rounded. It's once you get up into the top, you're less able to identify the apical meristem. Is it this one? Is it this one? Or is it even this one? It becomes harder to tell. Even in trees with strong central leaders, over time, they might become more rounded in appearance. So here's a pine tree, a juvenile pine that's shooting straight up, very strong central leader. This guy still has a strong central leader, but notice up here at the top, very top, it becomes a little less well-defined. So it can change over time. So delicate and changing balance of chemical signals that control metabolic processes of photosynthesis, respiration, and all biological functions. So again, you have to have this balance between all these hormones and compounds. Tree growth regulators impact each other and remain in a dynamic equilibrium. So dynamic means movement. So one will be a little bigger than or a little more available than the other given the time of year. Environmental stimuli and human stimuli trigger changes in regulator concentrations that stimulate changes in resource allocation, which result in changes to the tree's growth and development. Some other defense mechanisms that trees have, there are thick bark, thorns on the bark or thorns on the twigs, leaf hairs, the more fibrous the bottom side of the leaf, it actually forms a chemical barrier from things getting in. Thick cuticles can resist sunlight, cellular materials that resist decay and ingestion by insects, and we'll talk about phenols and some of the other compounds later. The production of chemicals that restrict insect feeding, pathogen infection, and decay. Now we're going to jump into compartmentalization quickly here. So compartmentalization of decay in trees. This is it's a theory that we use that was produced or founded by Dr. Alex Shigo. He was a forester who went out into the forest for a long period of time, intentionally wounding trees and then going back and looking at the tree's response to this wound over time. It's a very, very long, thorough longitudinal study. I actually had the opportunity and I worked with Dr. Alex Shigo for a few weeks, and it was quite a highlight of my career to get to work with this guy. He's one of the great minds of arboriculture. So what does this actually mean? It's, again, compartmentalization of decay in trees, CODIT. It's a process that allows trees to wall off decay. It's a process that allows us to describe this walling off. It limits the spread of discoloration and decay. After injury, reactions are triggered that cause the tree to form boundary areas around the wounded area. And this is where the CODIT model itself comes into play. There are each one of these responses we call a wall. So there's wall 1 through wall 4. So again, you have to put on your three-dimensional hat here to really see how this works. So wall 1 resists the vertical spread. So if I have a wound, if I have compression damage against the trunk, and I'm going to kill some of this tissue, well, that just like in a person, if you have a cut, you can have pathogens get into your cut. I don't want things to go all around inside of the tree. I don't want, say, here in Central Texas, oak wilt's a big deal. I don't want oak wilt to spread throughout the whole canopy, or anthracnose or bacterial leaf scorch, things like that. I don't want this to spread throughout the whole tree and kill the whole tree. So in order to stop that from moving around, I have to plug up all this vascular tissue. So remember, we had that tyloses that we talked about. It was typically a response to pruning. It can also be a response to injury. Remember, pruning is also an injury. So pruning is never helpful to health. Remember that, unless you're taking off deadwood. So you have this up and down movement in the tree. That's the most movement occurs in the up and down in the current year xylem tissue. So wall 1 is going to be to plug that from above and below, closing off the ends of the straw, trying to keep it contained inside of that straw. Next, we had the inward spread by depositing chemicals. It'll in the late wood itself, and we'll see this a little more in some other diagrams. Wall 3 is the lateral spread. So if I'm wounded here, and here's the inside of the tree, wall 1 is going to plug from here and from here. Wall 2 is going to be usually the previous year's growth increment. So that dense tissue that forms at the end of the season before it goes into dormancy and makes that ring is going to become the backside of the tissue that's being encapsulated. Wall 3, remember the ray cells. So the ray cells are going to inhibit the lateral spread of decay to keep it from going all the way around the trunk. Now, wall 4 is actually a physical barrier. It is new wood that is produced after the injury. So these are listed in their sort of order of durability and reaction. So this wall 1 can start to occur within 30 seconds to within a minute and a half or so of the injury. So if I drill a hole into the tree and I'm trying to introduce a chemical into the tree, I need to get that chemical in there in a hurry, or else the tree's going to plug the vascular tissue and my chemical's not going to move anywhere. So wall 1 happens fast, but it's also a fairly weak wall. It's easy to break down. Wall 2, that's that backside, so it takes it a little longer to get to the backside, but eventually it will get there, and it's a little stronger, but it's not super strong. Same thing with the ray cells. They are pretty durable, but remember, ray cells don't go very deep, so you might have to have overlapping ray cells as you go up or above or below the damage area to keep that lateral spread isolated. Then wall 4 is produced. If I had this compression wound, you'd come back and you would find this reaction tissue or wound wood that grows after the wounding. And that's very durable, very long lasting, and it's great at compartmentalizing. So here we can't see walls 1 here, right? Because wall 1's going up and down, and wall 2 has totally failed here. So the decay has gone all the way to the center of the tree. So what we have remaining is wall 3, which is these are the ray cells that inhibit the lateral spread going around the trunk. Also, year after year, that reaction tissue is going to continuously be produced, and it might even close over the wound, and eventually you might not even be able to tell that the tree was wounded there. That is incorporating the wound. Remember, trees do not heal. You need to take the word heal out of your vocabulary when it comes to trees' response to an injury. They cannot heal. They can close over a wound. A wound closes, a wound does not heal. So all of this wood that's produced after the injury, here's the wall 4. Notice that wall 4 can be seen all the way around here. It's pretty interesting. So it does form this sort of hard barrier layer there. Here's an example of the plugging of the wound above up here and below under it. So this would be wall 1. Trees over time, this is how that response would look. If that injury occurs in the trunk, then that decay can take over all the wood that was the size of the trunk. So eventually this decay inside of here can eventually decay out everything before the production of wall 4. So you get this big hollow column. If the injury extended all the way out to the outside of the bark and it was pretty big, then you can get stuff that looks like this. This is what that other photograph will look like over time. So all this tissue out here was produced after wall 4. This boundary layer is wall 4. When walls 1, 2, and 3 fail, decay can spread inside of the trunk and the hollow cavity is formed. The barrier wall 4 is strong chemically, but weak structurally. This is what they used to think. However, the new studies have shown that this material is actually pretty sturdy. And depending on how big the wound is compared to the total circumference of the tree, the production of wall 4 wood can actually be quite strong and be just as strong as can make up for the missing wood, but it's not as flexible. It's a little more brittle. The process that resists the spread of decay into new growth can also lead to shakes. So, if we went back and looked at this one, if there's a difference in wood density and all that sort of stuff as this tissue dries and shrinks or moves, eventually this could form a break, and so you could have these shakes or the splits in the bark that might form, or cracks. Quickly, we'll talk about tropical trees. As we said a while ago, these are going to occur closer to the equator. The anatomy is going to vary greatly. So the trees there close to the equator can look everything from these leaves that are as big as a car to leaves that are teeny tiny to, it's highly variable there. They may lack annual growth increments, again, because there's no seasonality to it. It's pretty much the same all year round at the equator. Many do not experience dormancy. Tropical trees may have extremely large foliage, flowers, or fruit. Some have very wide-spreading buttress roots. Rainforest soils or equatorial soils can be fairly thin. So the trees have these sort of above ground compression plates instead of underground compression plates. So you have these huge flares that come off of the main trunk. Some species even produce aerial roots that function in support. I'm sure you've all seen the stories about the banyan tree that's out in Hawaii that got burned up, and it's one of the largest trees in North America from a mass standpoint. Those produce lots of these aerial roots, and when those aerial roots hit the ground, they can actually become parts of the support system, and they can be responsible for gas exchange and propagation, vegetative propagation. New plants propagated in this manner are clones, because they'll be exactly like their parents. Tropical trees can also be identified by very rapid growth rates due to the favorable and continuous growing conditions. They are easy to transplant as long as you're transplanting them within their native ecology. Their wound closure and regrowth after injury, so their mechanism to cope with decay is to grow rapidly and outpace the amount of decay. When these trees are removed from their native ecology, you have challenges with their growth and health are likely to occur. So here in the temperate regions of the world, our rainforest plants are going to be our house plants, because the inside of your home stays relatively the same throughout the year. Yeah, so we get to palms towards the end here. So we're going to have a quick section on palms before our little longer break. Palms are monocots, or they are not dicots, and they are biologically very similar to a grass. So when they come out of the ground, they have one leaf blade coming out of the seed. They do not have a cambium layer or growth rings of xylem. So they still have xylem and they still have phloem, and they do have cambium, but the cambium is not in a continuous layer on the outside of the trunk under the bark. As a result, palms do not have what we would normally think of as bark. They have a coating on the outside, but it's really not bark. They have vascular bundles of phloem and xylem interspersed within the stem and sheathed in very strong fibrous tissues embedded in sort of this matrix of parenchyma cells. So here you can see, here's a sort of a monocot stem versus a dicot stem. These things are going to be scattered throughout all of the tissue, sort of distributed all throughout the palms. So the people that say, well, you can't prune palm. I mean, you have to prune palms with spikes, and spikes aren't really going to hurt the tree. Well, yes, they do, because every time you stick your spike in, you're spiking or damaging or killing instantly the vascular bundles associated with that part of the tree, because you're not jamming it into thick bark. You're punching it into this matrix of parenchyma cells, a living part of the tree. So there's not a dead outer coating. You see the stuff that sort of clings, the old leaf base, at the base of the old leaves can persist on the trunk and kind of look like bark, but it's really not bark. It's just part of the leaf that's still hanging in there. Stem develops all the vascular bundles it requires behind the primary bud as it grows. Remember how I said trees do not, or palms do not have secondary growth, so they don't get fatter the older they get. They are the same diameter coming out of the ground as they are at maturity. They don't get bigger and bigger, so there's no secondary growth, which means it has to sit there maybe for years, accumulating all those vascular bundles that it's going to want before it ever starts to shoot up. So you might go out into the woods in Florida or something, and you might see these green spikes coming out of the ground with a few palm fronds, but it's right on, the palm fronds are coming right out of the ground. Well, what it's doing is that's the phase of accumulating all these vascular bundles, and that's based on genetics. So once it reaches that maximum number that it needs, then the thing starts shooting up out of the ground. And so you get this sort of annual shoot growth that goes up and up and up, but it never gets bigger in diameter. So once it starts to shoot up, the trunk might be this big around. It could be, bigger around than my desk, or it could be, teeny tiny in diameter. Palms attain they obtain their maximum girth during this stage prior to beginning vertical elongation of the stem. Vascular bundles retain their conductivity throughout the life of the plant. So there's no apoplastic material. There's no intentionally dead heartwood inside of a palm. However, the stem is considered woody, although it's very different anatomically from the wood of a hardwood or a conifer. Cells in the stem thicken and strengthen as they age. So even though that exterior that I keep telling you is alive, it's a bunch of parenchyma matrix, the number and density of the fibers becomes greater over time towards the exterior of the palm's trunk. But that's still a living part of the tree. Cells in the stem thicken and strengthen as they age. So if you think about a palm, the oldest cells of a palm are right at the base where it comes out of the ground. The newest ones are up at the spike where it's continuously producing new leaves and that sort of thing. The stem is capable of storing starch because remember it's got this ton of parenchyma cells, so they can store an enormous amount of energy. Movement of water and minerals is not restricted to the outside of the trunk. So when we do things like trunk injections for different diseases, you can inject a tree in just one or two places and infuse the chemical, and that chemical itself diffuses throughout the vascular tissue as it moves up the tree. So the deeper you get it, the more concentrated those vascular bundles are, and so that's why you might see palm injectors being a little longer. We'll talk about those in a different section. Again, there's no secondary growth resulting in an increase in diameter over time. Palms do not have the ability to compartmentalize, so there is no annual growth increment. So there's not a way for it to respond so the CODIT model doesn't work in palms. Resistance to decay results from the strong, hard sheathing fibers of the vascular bundles. So those parenchyma cells and those sheathing fibers are very resistant to decay. So that's what's doing the job there. Palms only have one apical meristem. I keep referring to this spike. So if you look at a palm, you'll see this one sort of green, pointy piece going straight up in the air. That is the apical meristem. There's only one per tree. Once that one's gone, it's never reproduced. So the primary bud is protected by overlapping leaf bases of the emerging frond. So you have this spike, eventually a spike unfolds into a set of leaves. As it produces the set of leaves, a new spike is produced, and it's going to produce a set of leaves. And so you'll notice that palms have the spike, and they have these nice green leaves, and then some older green leaves, and older green leaves, and some yellowing leaves, and then some dead fronds as it goes all the way around the canopy. So it's constantly producing new ones from the apical meristem. Photosynthesis takes place only in the fronds. Fronds consist of a blade, a petiole, and a base. Leaves are produced sequentially, like we just suggested. Palms also have the largest leaves of any tree. The important takeaway here is if the bud is lost, the stem will die. If you lose that single apical meristem, the plant dies. When does that happen? Think about a hurricane. So if a hurricane comes and blows that off, or freeze damage freezes that apical meristem, but the rest of the fronds are alive. Every year people call and say, you know, I lost that spike in the top of the tree, but it still got lots of green leaves. It didn't die. And I'm like, yet. Because it will never produce new leaves after that. And those leaves can only live for a few years. So eventually all the leaves die and the whole plant dies. Palm flowers and fruit emerge in or below the crown from the leaf axils, the upper angle between the leaf and the stem, close to leaf scars or nodes. Flowers are clustered into large aggregates of many flowers. So you see these cluster of flowers. Flowers can be quite showy or they can be very inconspicuous. Flowers or inflorescences. Inflorescences, this is flowering, can be inconspicuous or it can be very showy. Some palms flower once in their whole life and then they die, but some produce, like date palms, produce a jillion dates every year with large numbers of fruit, and they live for years and years. The root system also is very different, as you can imagine, from the hardwood trees, because again, there's no secondary development. So notice in this picture, it just looks like a bunch of straws coming off the bottom. So that is the conductive tissue. So roots other than the primary seedling roots are adventitious. So if we piled a bunch of soil up here, it can actually start to produce more roots above this. You can also cut all these off and they grow back very quickly. Still, again, root development's at the base of the stem and the root initiation zone, which is the top 12 to 18 inches of the soil. Again, because this is where respiration can occur. So these roots still have to respire. They can be very densely packed the closer you get, and that's referred to as the root mat. So the closer you get to the trunk, the more dense the root system. If you cut these, they do grow back. So unlike a compression root, those large diameter roots, if you cut one of those off, it doesn't come back. And they do form mycorrhizal associations. So these things can extend out the height of the tree in all directions or even further, but they're the same diameter when they come off the trunk as they are out at the root tip. You know, there's some sample questions in here. I encourage everyone to go and look at these sample questions and try to answer them using the material, learning what you've used here. And it helps to get you sort of to critically think, and hopefully that's what we're accomplishing here today, is some critical thinking to help you with the exam to noodle through the different ways they might present the questions. So now we can take a little longer break.
All right. Does anybody have any questions before we head off on a 10-minute break? No, ma'am. All right, man. Y'all are quiet this morning.
I know it.
I know. All right, guys. 10-minute break. Let's head back at 11:35. Okay.
Right.
Thanks, guys.
Time to look for the other presentation.
I'll leave it up for you so you can do that.
Okay.
All right, everybody, are y'all back ready to get started? Awesome, awesome. Well, Rob, have at it.
All right, so probably are going to run just a little bit over, but we'll try to get in as much as we can. So if you have questions, please ask along the way. Otherwise, we might pick up the pace a little here. So this section is going to be about tree support and lightning protection. These are derived from those sets of standards that we talked about earlier, the ANSI A300s. So if you want the primary source, if you want to know a little bit more about the specifics of cabling, I suggest you go to that set of standards and purchase them for yourselves and read them and use them as a guideline when you're out in the field. So tree support is what we'll talk about first, and lightning protection afterwards. The benefits of cables, guys, and bracing rods or props. We'll talk about their why they're used and what their limitations are, the methods of developing and providing supplemental support, the techniques and materials used, what your responsibility as an arborist is going to be when you install these, and the benefits, and then at the end, the benefits of lightning protections and systems, the techniques and materials used with those as well, and how they're installed. So tree support. So junctions between codominant stems are considered the weakest portion of the tree. Remember our example of the Bradford pear earlier. Support may be needed for these defects for the trees with splits or decayed branch unions, multi-stem trees, or large branches that pose a threat or decent candidates. Tree support systems cannot eliminate hazard potential. So hazard is a technical word used in our industry. It is a legal word, a legal keyword. So be careful about how and when you apply that to your clients. So, but if something is a hazard, you're really never going to be able to change it from a hazard to a high risk or a lower risk of failure in terminology. We'll explain that in a different area. Trees must be assessed carefully and tree conditions considered closely before the installation of cables. You don't want to support this weak branch, this obvious weak branch up at the top, when you also have advanced decay organisms working at the base of the tree. Again, follow the standards. You can also follow standards in the ISA's best management practices, tree support systems, cabling, bracing, and guying. These best management practices are derived from the ANSI A300 specification. So there's lots of overlap in these supplemental resources. So cabling restricts the distance that branches can move in relation to each other. Bracing rods are used to reduce the risk of two or more leaders spreading further apart or moving sideways in relation to each other. Guying is the installation of a cable between a tree and some external point to provide that supplemental support or limit the movement. So if we limit the movement of limbs with weak connections, if it blows in the wind and it kind of does this, and we want it to not put so much stress down at the point of connection, we would install a cable so that it can't move any further away than some given amount that we allow it when we install the cable by how much slack or rigidity or tension the cable is under. We can also use it to support heavy limbs. So if you have very long branches and they're weighing down, we know at some point those things are going to break off. So we might install a cable to help that thing not break off. You also have trees with multiple leaders, like the Bradford pears that might have 7, 8, 9 stems, or it could be bifurcated, which is 2 stems, or trifurcated, 3 stems or more and more. Cables also can protect limbs or branches from heavy snow loads or winds. Possibly do it in every case. Cables should not be installed in trees that are not structurally sound. In other words, if it's about ready to fall over, don't worry about the stuff in the top, like we talked about. Trees with decay should be considered for removal rather than cabling. If you have a lot of decay, inside of the tree, there's not going to be much tree there to anchor your hardware. And so your anchor hardware can actually focus stress and pressure around that hardware and cause it to break. And so if you have a decayed area already, it could possibly break at the area where you install it. Before installing cables or braces in a tree, the tree should be properly pruned. So if you have a big full canopy, these branches that are wide and spreading, you don't want to put in a cable and have it nice and tight and then go through and remove all the weight off the tips, because then the branches will pick up and your cable will go slack. So be sure to prune them first so that the weight that you, or the tension that you put the cable under, remains fairly constant. It won't decrease after pruning. So there's three types of cable, the actual physical cable that they use. There's 7-stranded cable, so EHS and common grade are both 7-strand cable. 7 common grade galvanized. When you see the word common grade, that it means that it's malleable. Malleable means that it's easy to bend and to work with. You can move it around. EHS, which looks almost exactly like common grade, you won't know the difference until you try to bend it. So if you bend EHS, you're going to barely be able to flex it, and when you let it go, it's going to pop back to where it was. Whereas with common grade, if you bend it and let go of it, it's going to stay bent. The EHS is also very difficult to begin to bend, period. It's just hard stuff to work with. And then there's aircraft cable, so which is like the cable you might see on your bicycle, that woven stranded stuff, or it could be 19 strands of wrapped wire. So it comes in, most of it's that woven kind of wire. Aircraft cables are commonly used in 3/16 to 3/8 inch diameter. So this is fairly small to fairly large. This aircraft cable is used a lot in the UK and Australia. It's strong, it's flexible, but they are limited choices for attachments. There's also other places in Europe will use dynamic cabling, and we'll talk about that towards the end of this section. Anchor hardware. So anchor hardware is what anchors the cable to the tree or to the ground. Anchor hardware that passes through the limb is fastened on the opposite side as preferred. So you want to drill all the way through the trunk so that it passes through the whole tree. And then you put a washer and a nut down on this end, and then you have an eye on this side, and that eye is where you would wrap the cable through. That's the preferred method. That's called through hardware, as opposed to lag hardware. Some other hardware, you have eye bolts you can also have threaded rods and nuts that are commonly used. So in this next photograph, we'll see all the different types here. Get my laser pointer back up. Here we go. So you can have an eye here that is, you can see where this is called all thread or a threaded rod. So the advantage of the threaded rod is that you can make adjustments to it after it's in, or you can cut it to the specific length that you need and not waste material. This is a welded eye or an eye bolt. So the open or the round end is actually part of the thread. So here, once you cut off the end of this, you've wasted a lot of material. So it's not like a piece of all thread or threaded rod. So this one uses the threaded rod. Through hardware, again, going all the way through the tree, you want to drill a hole that is slightly bigger than the diameter of the threaded rod, because you want the rod to be able to easily, not super easily, but you want it to be able to move in and out of that hole without much resistance, because you're not using the threads. You're not using these threads to screw it into the wood. The only thing you're using the threads for are for the washer, the nut, and the eye part. So in order to pass it through all the way through the tree, you have to drill the hole a little larger than you would. So how much larger? 1/16 to 1/8 inch larger than the all thread, than the threaded rod is. The eye bolt or threaded rod is installed with a round washer and nut on the end. A long time ago, they used to use these diamond-shaped washers, and they thought the response to the tree was better, but over time, studies have shown that that's not so. The round ones produce the best reaction tissue. Remember, drilling a hole through the tree is wounding the tree, so you are going to have that CODIT response model exhibited here. If you have really thick bark, you want to chisel some of that away or cut it away a little bit. Here we have a Forstner bit that as it goes in, it chews away some of this bark. And then after we do that, we have that little pilot point, and then we can drill our hole through the rest of the way. But what we've done here is we've brought it down to the cambium without taking the cambium off. We might have taken a little bit of the phloem off. We're going to put a washer on there, and then we'll put a nut on it. And then since the cambium's still there, the cambium can grow that fourth wall of the CODIT model around the hardware that we're putting in. So the washer should be seated against the bark and the thick bark chiseled away. If you were doing a beech or a holly or a pear even, you probably would not do this. But if you're doing a cottonwood, you would definitely do this sort of thing. Let's see, did I miss anything? No. Okay, so this is the other type of dead end hardware. And this is a threaded or a lag hook. So these we're actually going to screw into the tree. Typically used on stuff smaller than 10 inches in diameter. Lag hooks or J hooks is a threaded J-shaped threaded anchor. So you can see how it looks like a J. So, and then these are fairly large. They're coarse looking threads. So you still have to drill a pilot hole to put these things in. But the difference is that this time you want the hole a little smaller so it can actually screw in. These things come in a left hand and a right-handed thread. So you want to make sure that one of them is left-handed and one of them is right-handed, so that at the end, when you're screwing this thing in and tightening up the system, that you're not untwisting, because it's a twisted cable. Remember, it's that 7-strand twisted cable. So you don't want to untwist all those strands. So you want the cable to move as one unit. So that's why you have a right hand and a left hand. If they were both the same, you would untwist the wire. When you're setting these things, you want the full length threaded into the wood. So you're going to screw it in. You might use, this guy's using a pair of pliers. You might also use a cable grip a cable helper, cabling tool or something like that fits in here and makes it easy to turn around. So as you're screwing this thing in, you don't want this end to get so close to the trunk that you start tearing off bark on your last screw or your last twist or so. So something else to see here that we'll come back and talk about, but notice this extra piece of metal that's inside of here. That's called a thimble. And these are called hand wraps. So you're going to unwind this thing strand by strand and wind it back around itself until this is your finished product. This is very strong. This this curve over the end of the thimble is called the bend radius. So from the center of this outward is the bend radius. The larger that bend radius is, the closer the cable is to meeting its tested strength. So the more strain you can put on it. The tighter that bend radius, the less you actually subtract holding power from the stated power of the cable. Again, this dead end hardware, this lag hardware is installed by pre-drilling a hole, but this time instead of the hole being larger, the hole is going to be 1/16 to 1/8 inch smaller than the lag, so that you can actually screw it in and it bites into the woody tissue. They should wind up so that when you finish screwing them in, that the J is straight up and down, not horizontal, and do it such that you don't injure the bark. Again, this works well on small trees or branches, no larger. Again, it was less than 10 inches is the standard. Here's some other things to be used. Heavy duty round washers are recommended. So heavy duty is grade 8 or better. Use these nice heavy duty things so that when you screw it down, it doesn't bend or deform the shape, that you're getting a uniform pressure against the trunk as you tighten them up, because you don't want to crush more tissue or deform the shape of this washer. Thimbles, here are the thimbles. The thimbles again are used to have that bend radius. As you can see, a thimble being used inside of this one. Dead end grips are used over thimbles to attach the EHS. The EHS is so strong that you can't pull the individual strands apart and start wrapping it around itself. It's not going to hold that way. You're going to have to use these dead end grips. So this stuff, you've probably seen them before and just didn't know what they were. They sort of look like these V-shaped things with a made out of cable itself, and they're flattened, and they usually have a coating on them. So you can wrap these things around, and they're sized, so you have to have the right diameter quick grip or dead end grip as you do cable. So the cable size and the grip have to match each other. This part wraps around and locks in place, and then you have another part that's sticking out, and then you wrap it around, and so this picture is not quite right, but once you wrap it around, it completes, so there are no gaps. So as you were to wrap this around, it would cover up or fill in these gaps. And it's super strong. And the older it gets, the stronger it gets up to a point. You can also use a come along and a cable grip. So a come along gives you mechanical advantage. So you have this long lever and the spool. So you could attach this into a strap, and the strap might go around the trunk. And this thing might go out towards the other branch that you're trying to pull the branch and the trunk together. You would attach the cable with the dead end hardware on one end, and then you tighten the cable up before you install it into the other end. And this is how you would do that. This grip actually holds onto the cable. So the cable runs through this little cam. Then this part, this little eye, is what would click in here. Or if there was a lot of distance between where you were doing this, you might have to use something else. But this connects to this part. As you pull in this direction, it rotates here and rotates here, and this cam moves and pinches on the cable itself. And so it holds onto that cable, grips the cable. So as you use the come along, it pulls the cable tight and pulls it towards the other end where you're going to put in the next anchor area. Then that way you put in all your anchor hardware, then you take your cable and you build the other end with the thimble and the wraps or the quick grip or whatever it is. And then after you have that in place, then there's a switch on here that you can flip and let the pressure off of it, and then the cable takes the full weight that the come along was using before, that the come along was experiencing before. This is a cable aid. This is one of those tools I talked about that helps to, other guy was pictured using a pair of pliers. You could use one of these things. It's got a little notch in here. These are soft aluminum, and they have curved edges and stuff so they don't tear up the bark as bad. Whenever we're drilling holes into the tree, we want to cut our way into the tree, not tear our way into the tree. The more precise your cut is, the better the tree will respond to the wound, and the more complete the wound wood will be in its reaction to being damaged. To do that, we use what's called a ship auger or a ship's auger. It's a drill bit, and they look a little different from a regular drill bit that you might see, the iconic looking one. These things are meant to be used at fairly low RPMs, revolutions per minute. They have a little pilot screw out here at the end that helps hold it into the wood and get it started. But these are hyper sharp. And so they cut their way in and make a very clean hole all the way through. So that's important for compartmentalization of the damage. We talked about that thimble. So here's the thimble. This is common grade cables. You're going to wrap it around the thimble and sort of hold it together, pull off one strand and begin to wrap it around to hold it in place. After you get the first wrap on, you can take your hand off of it and just unwrap until you've used all 7 strands, and eventually you have all 7 strands used up. And this gets smaller in diameter as you get towards the one standing part of the cable. These are used for the aircraft cable, this U-shaped stuff. They are used to, you'd go through, they wrap it back around, and then you put these U cables on. They should not be used to form terminations on cables greater than 1/8 of an inch in diameter. So if you're using bigger cable, you'd want to use those other end types of hardware, but this is what's used on a smaller cable. Install 3 clamps several inches apart with a U of the clamp over the short end of the cable. So this is the U of the U-bolt. So you want that part over the short end of the cable. Cable stops, now there's not a picture of one here, but cable stops are metal knobs affixed to the steel cable ends protruding through the drilled hole. So there is another form of cabling that you just pass the cable through that hole instead of the threaded rod, and on the other end you have this sort of cone piece and a cap, and it goes over the end and pulls into the tree, and there is no extra hardware. It's just that cable stop. So where do you actually install these things? So install the cable at least 2/3 the distance from the defect or the weak crotch to the ends of the limbs. So not 2/3 of the height of the tree, but 2/3 of the length from the point of defect. So here, if this crotch is the defect and this is the total length of the branch, we want to be 2/3 of the way up this branch and 2/3 of the way up this branch, and that's where we install the cable. We also want to envision sort of this line that bisects the defect, and we want this cable to be, this angle to be 90 degrees from here to here. It's important to have the cables pull in direct line with the hardware. So you don't want it to look like this. You don't want to have this nice horizontal piece of hardware pulling down at this direction, because you're going to lose efficiency. You're going to damage reaction wood that's developed over time, and you're going to focus all of the stress right here, and you're not going to distribute it on the backside, which is where we want the stress to be spread. So the way this should have looked is you should have drilled a hole at this angle so that the pull is direct. Or here, what you can get if you do, you can get that direct hole through if you get this angle right. So in this example, that angle would not have been right. So you're always going to pretty much drill the shortest path through the branch if you've bisected that point of defect. And so you can see that this one goes straight into the tree, and this one goes straight into the tree. But what's the problem that you see with both of these? I don't know if you noticed it or not, but they're J hooks wound up horizontal and not vertical. So you want these, another 90-degree twist on this one in either direction to make sure that you wind up with it vertical and not horizontal. The cable should be just taut. So you don't want the cable so tight that when you hit it, it sings a high note, but you also don't want it so loose that there's a lot of sag in it. If you have a very long run of cable, no matter how tight you make it, you're still going to have some sag in it. So just when we say just taut, that means that it's sufficient to do the job without excess movement or excess pressure pulling on it. If the cable is installed while the tree's in foliage, remember, prune it, and prune it before you install it, and you can put a little extra pressure on it if it's fully foliated, because after leaf drop, the leaf will pick up. But also after leaf drop, there's going to be less wind pressure on it, so sort of 6 one, half dozen of the other, but take into account that you don't really want it to go super slack after the leaves drop off. So here's some mechanical forces that might be at play in poor cable applications. So trees grow over time. They get taller and taller. Cables stay wherever you put them. Again, cells are fixed in that three-dimensional space. Whenever that cell is produced, it stays there forever, so it doesn't grow up with the tree or out with the tree. So wherever you put this cable, that's where it's going to stay. So as the tree gets taller and taller, when the top moves around, eventually the force changes to what you don't want, and it actually increases the problem down at the crotch you're trying to strengthen. So at the time of installation, this is a good install. The cable's nice and high in the tree. It might even be a little over 2/3 of the way up. But here we're gonna, and this is also what's called a dynamic cable, not a fixed cable, which we'll talk about in a second. So good versus bad placement. So people always say, well, what should I do over time? Should I install a new cable? Should I remove the old cable? So when the tree grows, you'll have to install new cables. You want one cable between two limbs or direct cabling. This is the most common type, so this direct cable. You might also incorporate triangular or box cable or pyramidal tray into your systems. Here's sort of a triangle. You can see the triangles, two triangles formed here. This would be a box installation where each branch is sort of held in place using another branch. So it keeps the cluster from moving apart. Now, these are not all at the same height in the tree. So we're looking down or looking straight up or straight down on these cable installations to see it this way, which with a direct one doesn't really come into play. But here, where you have multiple attachments to a single stem, remember we're drilling a hole through the trunk through each one of these. So this trunk's going to get 3 holes in it. And so we don't want, and there's going to be some decay associated with those 3 holes. So we want our cables to be at different heights so that that decay doesn't overlap. How much? So greater than the diameter of the tree. So if the tree is 10 inches in diameter, you want these 2 pieces of hardware more than 10 inches apart. So there will be, again, some decay associated with these. You don't want those 2 decay columns to line up with each other, because that can lead to more extensive decay in the future. You don't want the cables to rub as they go through the tree. You don't want it to rub against another big branch. Over time, you might see some sucker growth that comes up, so you might have to prune the sucker growth off to keep it from interfering with the cable. Only 1 cable should be attached to any 1 anchor. Can't tell you how many times I've gone out and seen like 4 or 5 cables that all come to 1 anchor point. They're pulling in all these different directions. That's not a good system. Anchors should not be installed into decayed wood where the amount of sound wood is less than 30% of the branch diameter. So if you have a large branch and it has a small column of decay inside of it, it's still okay to go ahead and put or install hardware into it. If you have massive amounts, so you've got a little thin shell wall and a big amount of decay, that's where you want to avoid using this through hardware or any type of hardware, because you're actually going to increase the risk of branch failure. Much of the holding power of these installs come after the fact. So it's the new wood that the tree grows on the outside, or that response tissue that incorporates the hardware adds to the strength of the hardware. What's your responsibility? Cables must be inspected annually. So you want to make sure to negotiate with your partner, with your either a partner company or with the homeowner or the landowner, the tree steward, to be able to get you out there to look at these every year and make adjustments if necessary. If they don't let you do this, then you need some type of release of liability to put the responsibility on the other person. Say, hey, I gave you the opportunity, you didn't take advantage of it, or if you don't take advantage of it, it's your problem, not mine. Over time, cables may need to be replaced in the tree as the tree grows taller and taller. So now we'll step into bracing. So these are steel rods installed into the tree to reinforce a weak or split crotch or to strengthen decayed areas. They are used in conjunction with cables. They are lag threaded rods, wider and deeper threads. That means like it bites in, or they can be machine threaded rods, which is the narrow one that passes through. So notice that it says used in conjunction with cables. You cannot install bracing material without also installing cables. You can install cables without bracing, but not bracing without cables. Lag threaded rods, just like in the J hooks, you're going to drill a slightly smaller hole so that the lag thread actually bites into it. You're actually screwing the 2 parts back together. The advantage is that the threaded rod itself provides the support. Machine threaded rods, these have the very fine threads on them. These you're going to drill all the way through, and the anchor hardware is going to be on the backside. And since you're going all the way through, you want your hole to be 1/16, 1/8 inch larger than the rod. A single rod should be installed above the crotch at a distance of about 1 to 2 times the branch diameter. So you want it up here. Cut off any excess rod beyond the nuts. So you're going to cut them off here and here. Then you're going to peen the end of the rod. Peening means you're going to beat it. So if you knock the hammer onto the thing, it makes a sound like a peening sound, but you're crushing the thread so that the nut cannot back off. Here's an example of what not to do. I was driving along. You know, you might not notice it as you're driving from the street, but you stop and you get a little closer look at it. You'll see that not only he's done like as many bad things as you can possibly do here. The angles, notice the angles are not in direct pull. He's also used square washers instead of round washers. He did not cut off the excess material and peen the end of it. He didn't even use a cable. He used a buckle here to hold it all together so that he can twist it and make it tighter, I guess. And then notice this is a pretty serious split. And there's likely a house right here. There was a house because I remember it. And so you don't want this thing splitting and destroying your house. The tree was pretty high risk, but he did this on his own accord. So he takes all the responsibility for it. This would not be a tree I would sell. I would probably just sell removal right there. So dynamic cables, if you remember back to when we talked about the word dynamic, it means movement. So these dynamic cables allow for more tree sway. They don't just like a cable is going to pop and it's going to move out and then stop at the end of the cable. This thing will come down and then it slows down before it comes to a stop. So they're German-made systems called the Cobra. The US make one called the Tree Save. But it wraps around the tree, so there's no hardware that you're drilling into the tree. So these definitely need adjustment every couple of years or so. This is tubular webbing, so it works a lot like one of those finger puzzles where you stick your fingers in it, and then when the harder you try to pull them apart, the tighter it grips your fingers. So that's what's happening here. And there's a little swollen area in the middle that has a snubber built into it. So this is a thick rubber cylinder that you stuff inside so that when the tree pulls apart, it's squishing down on that snubber, and that's what allows it to slow down. That's where the dynamic part comes from. These things are easy to adjust. They last for years. They're easy to move around, but again, you have to have them inspected annually. Guying, you can use. Guying means that you're going to attach your hardware. One part's going to be attached to the tree somehow, and the other is an external point. The external point can be another tree. It can be a ground anchor. It can be a rock, anything on the ground or off of the main target tree. This is guying, an external support. Again, this is going to reduce tree movement. Guy wires should be attached to the tree at a height at or above the midpoint or 2/3 the height of the tree. The higher you get, the more leverage you're going to get on the tree. So anywhere from the midpoint up to 2/3. And again, these trees get taller and taller. So as they do so, you might have to install additional cables on these. Ground anchor should be placed no closer to the trunk than 2/3 the distance from the ground to the lowest attachment in the tree. Another way of support is what we call propping. Propping is the installation of a rigid structure between the ground and a branch or a trunk to provide support. They are used under branches or leaning trees that are nearly horizontal or downward growing. This is used a lot in Europe, Australia, New Zealand. They have these props. This is a dynamic prop, so these things actually move around. You can see all those other little cables inside that move as the tree sways in the wind.